very warm good morning to all distinguished dignitaries, respected guests, eminent speakers, and dear participants. On behalf of the School of Medical and Allied Sciences, K.R. Mangalam University, it is my privilege to welcome each one of you to the day 2 of the two-day international conference on Pharma and Allied Sciences in the age of AI, innovations, and IPR strategy, PASAI 2026. This prestigious conference is proudly funded by DSIR, ANRF, Haryana State Council for Science and Technology, and is sponsored by SD Biosensor Healthcare Private Limited, Getwill, Advent Naturals, and RP Northland Institute, and bringing together brilliant minds shaping the future of healthcare and innovation. It is my great honor to officially inaugurate day 2 of this international conference by welcoming our distinguished guest of honor on the dais. I feel privileged to invite our esteemed dignitaries to kindly grace the dais. Dr. Anup Singh, Scientist E, DSIR, Ministry of Science and Technology, Government of India. I request sir to please grace the dais. Please give a round of applause. Now I request Professor Dr. Sanjay Rajpal, General Manager, Ipsum India Private Limited, to grace the dais. I would also like to respectfully invite Professor Dr. Hema Chaudhary, Dean School of Medical and Allied Sciences, to kindly grace the dais. Thank you, sir. Thank you, ma'am. We are truly honored by your presence. We now move to the floral felicitation of our esteemed guest as a token of our heartfelt gratitude and respect. May I request Dean SMAS to kindly felicitate our esteemed guest of honor, Dr. Anup Singh, as a mark of our warm welcome. Now I request Dean SMAS to kindly present floral bouquet to our distinguished guest of honor, Professor Dr. Sanjay Rajpal. Thank you, sir. Thank you, ma'am. Now I take the honor of introducing our guest of honor, Dr. Anup Singh. Dr. Anup Singh is a scientist E at Department of Scientific and Industrial Research, DSIR, Ministry of Science and Technology, Government of India. Before joining DSIR in 2013, he worked at Technical University of Denmark, the Energy and Resource Institute, New Delhi, Banaras Hindu University, Varanasi, and VPS Purvanchal University, Jaunpur. He has visited several European countries and has published more than 50 research articles in various scientific journals, and having H index of 39, I-10 index of 67, and citation more than 12,000. For the past 3 consecutive years, Stanford University has ranked Dr. Singh as one of the top 2% scientists in the world for excellence in scientific research in the field of energy, environmental sciences, and enabling strategic technologies. Respected sir, I welcome you for addressing our esteemed gathering. So please.
Distinguished guests, eminent speaker, faculty members, researchers, distinguished delegates, students, and members of the community. Good morning to all of you. It's my privilege and for the second day of the two-day international conference on pharma and allied sciences in the age of AI, innovations and IPR strategy, organized by the School of Medical and Allied Sciences, Kerala University. At the outset, I extend my heartfelt congratulations to the school, the university and the entire organizing team for successfully conducting the important and timely conference. And first of all, I want to introduce my department. So we are from a Department of Scientific and Industrial Research. This department comes under the Ministry of Science and Technology. Other than BSc, Ministry of Science and Technology has two different other departments: DST, Department of Science and Technology, and DBT, Department of Biotechnology. So we are dealing with basically industrial research. And the major program of PSIR is to provide regulatory like recognition for research activities to industrial in-house R&D units, scientific and industrial organizations, silos like this university, and PFRI, public funded research institutes like IITs and other state or central government funded institutions. So other than that, we also have some funding programs like events, we have conferences, workshops, etc. We are providing partial support. Other than that, we also have a program TDUPW, Technology Development and Utilization Program for Women. So under this, we are providing financial support for training women in rural areas or in say weaker section area. Other than that, we also have PACE program where academic and industrial units can put together their proposal and we are supporting them. So coming to the theme, so the theme of this conference is highly relevant in the present era: artificial intelligence, machine learning, data analytics, and digital technologies are rapidly transforming pharmaceutical science and healthcare. Not this in every area, AI is very important nowadays. Today, AI is playing an important role in drug discovery, molecular modeling, formulation development, disease diagnostic, clinical analysis, personalized medicine, and digital health. These tools are helping researchers reduce time, cost, and repeated experimental efforts while improving the quality and effectiveness of care solutions. I am happy to note that this conference has brought together pharma, allied sciences, artificial intelligence, innovation, and IPR on one common platform. Such interdisciplinary conferences are essential because the challenges of modern healthcare cannot be solved by one discipline alone. Scientists, pharmaceutical, clinicians, engineers, data scientists, patent professionals, industrial experts, policymakers, and entrepreneurship must work together to create meaningful and practical solutions. For young researchers and students, I would like to say that this is a very exciting time to enter the world of science and innovation. You have access to advanced tools, global knowledge, and new opportunities. But true innovation begins with identifying real problems. Your research should aim to provide practical solutions related to patent care, affordability, formulation stability, diagnosis, treatment compliance, and healthcare accessibility. I also encourage faculty members and students to develop an IP-oriented research mindset. Before publishing your research, always consider whether your work has patentability, commercial potential, or societal application. Publications are important, but patents, prototypes, startups, and technology transfer are equally important for building a strong innovation ecosystem. As India moves towards the vision of Viksit Bharat, universities and research institutions have a major responsibility to promote innovation, entrepreneurship, and industry collaboration. Our focus should be on affordable, accessible, and technology technology-driven healthcare solution for the society. I once again congratulate K.R. University for organizing committee, speakers, participants, sponsors, and volunteers for making this conference successful. I am confident that this conference will lead to new ideas, collaborations, publications, patents, and future innovations in pharma and allied sciences. Thank you for inviting me and giving me this opportunity to share my thoughts. I wish the conference grand success. Thank you. Jai Hind.
Thank you, sir, for your words of wisdom. Now I take the privilege of introducing our next guest of honor, Professor Dr. Sanjay Rajpal. Dr. Sanjay Rajpal is a distinguished healthcare professional and author with over 2.5 decades of experience in healthcare industry. He currently serves as an honorable professor at School of Paramedical and Allied Health Sciences, Centurion University of Technology and Management, and is also a country manager of Pearsoned India Private Limited, overseeing operations in India, Sri Lanka, Bangladesh and Nepal. Dr. Rajpal holds a doctorate in public health management, a postgraduate diploma in public health management. He is also a certified life coach for International Coaching Federation. With his vast expertise, he has mentored numerous young professionals and newcomers in the field of diabetes education and healthcare sector. In addition to his corporate and academic contribution, Dr. Rajpal is a prolific author, having written and co-authored 8 widely acclaimed books. His work spans across healthcare, management, communication, and public reflection, reflecting his deep commitment to knowledge sharing and professional development. Respected sir, it is my honor to welcome you to address our distinguished gathering.
Good morning. Namaste. Am I audible? Last minute of batting, can they hear me? Okay, very good. So first of all, respected Dr. Noop Singh, very nicely as a visionary, he set the context of the whole day as well, I think so, like a Viksit Bharat. Thank you to our Dean Madam and Hima Chaudhary Ma'am. Thank you very much for introducing today morning the whole institution and the journey and the vision you have. So I'm really impressed. Before the man has read very nice my CV as well, but just give me a short introduction from where I come from. I had one Swiss healthcare organization based in Delhi only, and this organization has based in Switzerland, and we make self-injecting pens like these. And these pens, any idea how use होते हैं? Please, my request is, ma'am, can I ask you some questions as well? Is it okay? Please, I need your permission because you are. Whatever I will speak, I may ask you some questions as well. The purpose is dialogue. सिर्फ मैं बोल के चला जाऊँ फिर आपको लगे भाई session is over now. And how much time sir I have? Why sir? How much time I have? I will stick to that time. Whatever you say, I will do it. That's very kind, but still. So this company is an organization. These pens are used for definitely for diabetes one, and besides diabetes, even during pregnancy, follicle stimulating hormone, and during arthritis, PTH, parathyroid hormone for cancer treatment as well. So because you all are from healthcare, allied sciences, and these pens comes in reusable as well as disposable pens. And now because of how artificial intelligence gets connected with these pens, the new model which will come, because compliance of something is a big challenge. Nobody reads the IFU and everybody just wants to use injection and go. And the next pen we are launching, that how that pen will read that अगर patient ने या user ने proper dose नहीं है, then red light will come. And whole data, if somebody's father is taking insulin and he's not taking a proper insulin, the doctor can also see on the mobile phone और अगर उनकी डॉटर है, even she can see कि पापा ने proper dose लिया है नहीं. So this is how artificial intelligence, how as a healthcare professionals, how technology will affect our profession. Because we all talk about accessibility, affordability. So these are the things how AI is going to bring the change. And definitely these pens, so it looks like, and these pens require needles. These are not needleless, but needles are always there. So this is where we come from. So this is the background of our company. And besides this, can I have 2 volunteers? Could be 2 students, please. Any of the 2 volunteers. कोई भी आ जाए. तो मैं देखिए, either you come on your own, or I will pick on my own. Up to you. Because either victims you call it, because purpose is a dialogue. कोई तो बच्चे आते. And very good, child. Are you also very good? Very good. You also. Yes, very nice. Give them a round of applause, because if some speaker calls, it's very heartbeat goes up. Why me? मुझे क्यों बुला लिया? But I appreciate. आप अपने आप आए, very good. And you all will get a special prize from my side. Ma'am, I wish to, I will give you some prizes, right? And again, my request will be that I do, because you have a very distinguished speakers. I have read the complete, very nice agenda. Just let me share first one story. And before I share the story, let me ask you. How this will change the subject. Don't give them the card, let them pull one card. Even I will pull one card. Even give to ma'am as well. Give ma'am as well. Sir. Even I will pull one card. Any card. And how do you see that whole thing works? Yes, yes. Let ma'am pull one card. I already give it to sir. Sorry, kill it. Because the purpose of the whole Congress is that there should be a value addition. Even if I have come all the way 2 hours drive, even I will not feel happy if there is no value addition. Everybody has got one card, and every card. Every card has got one value inside. Let me start with myself. My value which has come is balance. And now, how are you going to use this value? Either in today's whole day. Anybody volunteer would like to answer? Then आपका card है. What's your value? Purity. Purity, yes. Can you please share?
In the conference, I will do whatever the work with my wholehearted, with the
Beautiful, beautiful.
and
Very good.
yeah, I got this conference will become a great success, and whomever is so coming here to get the knowledge and inspiring the thoughts given by our keynote speakers, our guest of honors, and whomever so is coming to get the knowledge from this conference in the age of AI.
Wonderful, man. Give her a round of applause. Anybody who does good, and there is no wrong answer. कोई भी wrong answer नहीं है, don't feel. And can I have one volunteer? One, हाँ जी आप बताओ बच्चे आते हैं. Yes, yes. See, anybody who will avoid the eye contact, उसको आना ही पड़ेगा फिर. So please don't avoid it. Very good. Give her a round of applause. Very good. जोर से बोलो. I always believe if you clap for somebody today, tomorrow somebody will clap for you. So always have this mission. So do not hesitate. I know mobile phones are important, but for a few seconds you can keep aside. Give a round, once again, round of applause. Whole Mangla Bureau बच्चे आगे, बच्चे come here. आपका नाम क्या है?
तान्या सैनी।
तानिया से। तानिया बच्चे आपकी क्या वैल्यू है? क्या वैल्यू है? Forgiveness, beautiful. तो आप बताइए, आज के पूरे दिन में how are you going to this value? Yes, very good.
I will try to forgive anyone who have anything with me in past or in future.
Beautiful, beautiful. Very give her a round of applause. Very good. Thank you, Tanya Sani. Any boys भी आ जाए please. Tanya, very good answer. Give please. आगे boss, very good. What's your very good. What's your name, boss? आपका नाम?
My name is Sharad. Sharad.
शरद, आपकी वैल्यू क्या है?
Equality.
Equality. Very good. How you are going to use equality?
As a volunteer in international conference, I will equally treat everyone, respect everyone.
Beautiful. Wonderful. Give us Sharad a big round of applause. So I cannot go to everyone now because that you can use later. Last, I will ask the madam. Mercy. Very good. So ma'am, how you are going to use this value? Give her a ma'am. Because ultimately, give ma'am a mic. You see, the card has come, ma'am has pulled the card. We have not given any partiality. Ma'am ने card pull किया.
So I request that if anything we have not reached up to your expectations, then the mercy should come in mind.
So thank you very much. My request is the purpose of the whole exercise is I usually when I have a big meeting, I have some big presentation. Even I use card, okay, Sanjay Rajpal. I will follow this value today. And how it works, you see how it changes your whole paradigm. And my request to especially to students, people who are in the front rows, they are very experienced, professors, researchers, running the organizations. मेरी आपसे request है try to take the best value of this organization. You are from कहाँ से भी आप लोग हैं? Delhi, Haryana, Punjab, wherever states you come from. If the institution has done such a big effort to organize an event, to bring such experienced people, मेरी आपसे भी request है. Do not just sit here for because this time will never come back. Please, promise. Another, so I will just share one story. And because I know that I don't want organizers to give me a red flag, one story and then I will conclude. How many of you heard the name of Starbucks? Please raise your hands. ठीक है? आपने सुना है Starbucks? Let me share one story. And this story really touches me. When Starbucks started, it was not doing well. It was not doing well. And then today it is everywhere in the world. It's a multi-billion-dollar chain with at least, I think, 40,000 plus outlets. And it was not doing well. And then what happened that management decided, we will close it out. There's no point. Profitability is not coming. ROI is not coming. We will close it out. Then one new CEO was hired. Some gentleman, you have to close. Mandate was given that in next 6 months, you have to close it. All employees to be given pink slip and all lease agreements to be finished because not doing well. The gentleman who joined and his message, fine, 6 months are there, and he said, fine, Starbucks may even single person will come in the whole day. He or she should go with an experience. Right from entry the door, whether they take cappuccino, Americano, latte, whatever they take, hot chocolate, whatever, they should go with an experience. What happens? Fine, we have 6 months and then we have to close down. Board of directors mentioned isko band karna hai. ठीक है जी? Then what happened that even every employee was given the mandate, जो भी enter होगा, 1 घंटा बैठे, 2 घंटा बैठे, they should go with an experience. Same, one person come in the whole day or two, not many. But because of this philosophy of the mindset that all whatever outlets उन दिनों थे 10, 10, 20, but every visitor was given a feeling something that you are special. And this campaign जो उन्होंने नाम दिया था move the buttons. ऐसा नहीं है कि आप काउंटर पे आए, आपने बिलिंग करवाई, and then you went back, and you know. Even till the time the person leaves, opening the door, how to treat them. What happened? That message started going all over. That this is the real place. 5 months passed. Same board of directors, those who said isko band karna hai, they told the CEO that they started numbers going up. Value going up. They said, "Nahi, please continue for more 6 months." And now today is the history. The lesson of the story is come out of comfort zone. Because this time will never come back. You are 20s, 19, 20, 21, 22. आपको लगता होगा conference में हम आ गए. Sir has organized, so many speakers have come. Please interact. Please talk to them. बात कीजिए उनसे. Do not hesitate. And I can promise you, none of the speaker आप किसी से भी बात करेंगे, nobody will shoot away with you. Everybody, I can, any किसी से भी आप बात करेंगे, boss, this is a real big effort. This is a real big effort, attending a congress, attending a 2 days conference with such an esteemed people. अगर आप इसका फायदा नहीं उठाएंगे, तो सिर्फ अपने फ्रेंड्स के साथ आप बैठिए, बात कीजिए. You please talk to your friends, no problem. But still, make your orbit bigger. Promise? Yes or no? Yes. Promise आपसे नहीं आवाज. Yes. Yes, sir. Whatever time is left today, till 5 p.m. till 6 p.m. My request is that today should be better than yesterday. And ultimately, man will be happy that if you get the better outcomes. I ask, and I would like to give madam. I will make you the jury. Shri, we have 2 books. Ma'am, 2 of the students, those who participate, those who are keep on smiling, looking less on their mobile phones. You make 2 juries. These are 2 books which are authored by me. It's my first book, and it's all about etiquettes. It's my first book. At the end of the day, you just send me the pictures that who got the best. But my request is, boss, we are now seniors, so we have to give back. These books मैं आपके लिए छोड़ के जा रहा हूँ. So you just, ma'am has specially installed maybe 10 cameras inside, so she will hold, check, and then hand over. So without taking much of your time, whole purpose of the event of this congress is get best out of it. ठीक है जी, करेंगे, and keep smiling. Don't feel pressure. My God, we have to sit. We are very fortunate people. Outside it is very hot, but we are very fortunate that we are getting air conditions. All air conditions are very nicely working. We have a good water, everything. So have gratitude. जो आपको मिल रहा है ना, इसके लिए feel blessed. Sometimes this feeling comes, ये तो हमारा entitlement है. This attitude comes. But my request is, there are many young students like you as well. There are many institutions as well, those who do not have this kind of infrastructure. आप एक अच्छे fortunate बच्चे भी हैं. Thanks to your parents, thanks to your university management, thanks to your faculty. So try to take best advantage of this. So I wish you all the best for a productive, for a value-added congress. Have a good day. Thank you very much.
Thank you, sir, for sharing your valuable insights and expertise with all of us. We shall now proceed with the felicitation ceremony to honor and express our heartfelt gratitude to our distinguished guests for gracing this occasion with their esteemed presence. May I now request Dean SMAs to kindly come forward and facilitate our guest of honor, Dr. Anup Singh, with a memento in recognition of this valuable presence. Now I request Dean SMAs to kindly felicitate our next guest of honor, Professor Dr. Sanjay Rajpal, as a token of our heartfelt appreciation. Again, I'd like to thank all esteemed dignitaries for gracing the dais and making this session truly meaningful. As we now move forward to the technical session, I respectfully request our dignitaries to kindly take their seats and continue to bless the proceedings with their presence. The first technical session of day 2 now begins. This session marks the continuation of our scientific journey where ideas transform into innovation and research meets real-world impact. We are privileged to have this session guided by esteemed expert in the field. I'd like to introduce our respected session chair for this segment, Dr. Shikha Sharma, who is working as an associate professor at HOD, Department of Pharmaceutical Sciences, KC Group of Institute, Himachal Pradesh, as our first session chair. Ma'am, please raise the dice. I request ma'am to please invite our first plenary speaker, Professor Dr. David now.
Good morning, everyone. Good morning, all the respected dignitaries. It's given me an immense pleasure to introduce Professor Dr. Kevin Now, who is working as Dean, Faculty of Applied Health Sciences, University, Bangkok, Thailand. Dr. Tavin has focused his research on natural products for neurodegeneration and anti-aging at Chulalongkorn University with more than 11,150 citations and H-index 36. He has successfully upgraded the research quality in this field as evidenced by at least 209 publications. His research team employed a variety of experimental models such as cell culture, yeast, and animal models to elucidate the mechanisms of natural products to fight against neurotoxicity, neuroinflammation, neurodegeneration, and aging. With this, I would like to invite Sir on the stage and please share your knowledge and your words of wisdom with us.
Thank you very much for the kind introduction.
Thank you, sir.
Good morning, namaskar. So today I'd like to share the findings of my work. I'm not sure if I can use the my work that I have been working in this field for over 15 years. And first, I'd like to thank the organizing committee, the Dean, as well as Dr. Mohin for this kind of invitation. I'm really happy to be here, and hopefully I can briefly tell you about my research finding this morning. My background, actually, I got a PhD from Virginia Commonwealth University in Virginia, USA, and I studied the control of the lipid exchange. That lipid called galactocerebrosidase. Galactocerebrosidase is very important lipid because it is synthesized and also be a key component in the brain, particularly in central nervous system and peripheral nervous system to make the myelin. So without this lipid, myelin synthesis would not be possible. I'm sure that you know the function of myelin, which really controls the brain function. So this is very important. So since then, I realized that the lipid is very important, really critical, because we know previously that the person who discovered DNA is superhero and really well known. And protein also, whoever discovered protein and can do the analysis of the protein, those people would be really well recognized. But actually lipid is very important, particularly in the brain where we have the lipid of 70%, only 30% of the protein. So in the brain, the lipid is the main component. It's the main component. Whereas in other cell of the body, we have less lipid but high protein level composition. So this is very important at that time. And then later, lipids still play a role. For this paper, as you can see, lipid in the mitochondria is very important, but it has adverse effect because lipid in mitochondria can induce the oxidation and finally generate the free radicals and make the adverse effect to the cells, which is not good. So lipid actually have pro and con effect to the body, to the cells. Back to Thailand, my country, we have a very critical problem where we have high number of people, population with aging. I mean, the age at least 60 years old, we have over 21% already. So this is high impact of the aging society. And this pyramid shape of the population has been changed a lot, where we have less children born and we have high number of aging. And even now the labor supply also, we have a big problem also. So we have to import the labor supply from the neighboring countries like Laos, Cambodia, Myanmar, for example. And as you can see in this picture, the slide, about 10 years ago, we are in the top 10 of the population aging in the world actually. But next 9 years or next 10 years, the number of aging people will be double. So this is very critical problem. So Thailand government now try to enforce a policy that whoever in the academia can do the research to solve the problem with aging. We make it the fund to support the research much more easier. So that's why I try to adapt the knowledge of neuroscience, lipid, and then herbal medicine. Try to find the herbal medicine, drug discovery, natural product study to get the new possibility, new drug or new nutrients to solve the problem with aging. As you can see here, when people has aging, high number of people with age, we may face many problems like Alzheimer's disease, diabetes, cancer, as well as cardiovascular disease. But my lab actually focus on Alzheimer's disease only. That's why I want to show you some data with Alzheimer's disease study. And this data actually in line with the discovery in Singapore. In the last 2 years, when we check the number of the patients who go to the hospital, the last 2 years, they went to the hospital because of three H disease. Number one, hypertension. The second one, hyperglycemia. The third one, hyperlipidemia. Hyperlipidemia means high fat, high cholesterol, something like that. Hyperglycemia meaning that people may have the risk or high risk for diabetes, for example. and hypertension. It could be everything if people have hypertension. So we call 3H. This is the very high population in Thailand who have these causes and they went to the hospital. So if we can do in the future, we can reduce the number of the people who are having a risk with these 3H diseases. And this is the campus of my university, Chulalongkorn University, which is located in the heart of Bangkok, Thailand. It looks like the palace or the temple, but no, it is the university. The king, about 100 years ago, the king gave the land to the university. So we are really like the university who are the family member of the king. So that's why it looks like the palace, just like this. And we have the symbol, everything in the university with the logo to the royal family as well. Even the royal family members also they send the children to study in this university. So Chulalongkorn University is the name of the king in the previous Thai history actually. And you look at the map of Thailand from the north to the south, the west to the east, we are rich of natural products. We have a million species of natural products. So I could go to the farm, go to the forest and collect the natural product and study some of them. And I want to tell you one thing, over 100 years, the scientists have attempted to discover the drug for Alzheimer's disease, but still not here nowadays. It's very hard to do, over 100 years. So I'm not sure actually how far we need to go to discover the drug for Alzheimer's. But the best way is that try to prevent the disease rather than be the patient. Because when you be the patient with Alzheimer's disease, your memory will be lost. Some brain cell will be lost also. It's very hard to reverse. So once you get a disease, reversal is impossible. So prevent the disease better than curing the disease. So that's the point. Then I should find natural product, the fruit or the vegetables that you eat that prevent the disease. So I would like to share some of the story. And looking about the disease, like Alzheimer's disease, we know that some pathway, signaling pathway or the marker to show that you are Alzheimer's disease patient. They will look at the brain. The brain may have high amyloid beta, high tau of Alzheimer's disease. If we know the hallmark of the disease, then we may get some drug, new drug, fruit, vegetable, given to the people or the animal model, and then those hallmarks of disease may be reduced. One of the hallmarks that's very important to study Alzheimer's disease is still mitochondria, which is the powerhouse of the cell. So my lab really look at the mitochondria. For this picture, you can see that the healthy mitochondria look really nice, like normal mitochondria. But the Alzheimer's disease, which is AD mitochondria, look just like that. They have the big vacuole in the cell, so large vacuole, empty vacuole in the cell. So that reflects the Alzheimer's disease model. But very hard to find the drug for Alzheimer's disease because some drug we cannot test in the human directly. We have to test in the animal. Do you think the big animal or small animal better to translate to human when we get the finding? Should we use the big animal or small animal to resemble the human? Big or small? Bigger is better, like monkey, rather than rat or mouse. Big animal, because they may almost similar to human. They have number of genes almost similar to human, for example. So the elephant would be the best. But I can show you why elephant should be the best. But in my lab we cannot culture or we cannot raise elephant because it's too heavy to bring to the lab. But this is just a story thinking about elephant. Elephant actually only 5% of them die due to cancer, whereas human about 11 to 25% die to cancer. This may be because of genetics. Elephant has the cancer suppressor gene. Elephant had been published long time ago. This is the real story and compared to the human, also published in JAMA a few years ago. So that's the real story. But now I'd like to show you the study done in my lab about the fruit, vegetables that we studied, many of them, particularly the colorful vegetables and fruits like this, like in the picture. And if you go to Thailand, you will see this picture, the floating market, where you can find variety of the fruit, vegetables along the river. So they call floating markets. In Bangkok or nearby Bangkok also we can find this picture anyway. So based on the season, you can find different types of fruits. But keep in mind that to do the research, we have to focus not only publication, because this guy who got the Nobel Prize in economy, he said that innovation may be really important rather than publication. So try to do research and then we eventually can apply for human use. And also, don't forget that we need to concern about SDG as well. So you need to know about SDG anyway. I won't talk a lot about SDG here, but you can find it a little later. That's the picture. SDG goals very important, like health and well-being, the story I'm sharing with you today. And the story that I like to take as an example that we are really successful with the study, this is the passion story. His name is Pasindora Idolis. We took them to the lab. We know that some of activity known so far about this fruit, it can mop out the harmful free radical, it can prevent constipation, for example. But my lab studied neuroscience. We want to know whether or not the passion fruit can prevent Alzheimer's disease or can kill the Alzheimer's disease. So we took them to the lab. We studied every part of the passion fruit, but we found only the inner part with white sheet. The white sheet in the part of this passion fruit that can prevent the disease in the lab. This is the extraction process. We extract the passion fruit with different methods, different solvents, for example. And then we know that hallmark of neurodegeneration, including the amyloid beta pathology, the tau pathology. So if you look at the neuronal cells, like I showed you, the neuronal cell with the myelin, for example, each location represents each hallmark of the disease, from axon to dendrite, for example, from many branches that you can see here. These are the name of the models that I use in the lab. I use C. elegans, cell lines, drug bioinformatic or in silico analysis, and I also use the yeast model system. Today I like to show you the study in cell line, which is brain cell, and the C. elegans. The C. elegans is the worm, really small worm. The size of the C. elegans like 1 millimeter, and there are a variety of strains of the C. elegans. I will show you the picture of C. elegans anyway. It's just the worm, very small worm. But this worm has 3 complete systems in the organism. Number 1, they have the digestive system, meaning they can eat. The second one, they have the brain system, the simple brain system. We can study Alzheimer's disease. And the third one, the worm has 3 reproductive systems. So they can produce the eggs, and the new worms, for example. And in the cell line, we know that once the cell got incubated with the glutamate, the glutamate is the neurotransmitter. Once you have accumulation of neurotransmitter, eventually the cell will be dying. But with the extract of the passion fruit, the cell can survive even in the presence of high glutamate level. And This is the picture of the silicon, and you can see the life cycle of the silicon, it's a small one. Initially, I could not do the silicon worm culture in my lab until I met the professor from Germany, Professor Wing, who went to Thailand and gave a talk. I connected with him. And then since then, I realized that networking in the conference like this is very important. So if you don't get any message today, at least you do know people and then try to connect with them. So networking, very important for the future collaboration. And then Dr. Prashant in the picture, who's Indian postdoc from Allahabad University. When he got the PhD from Allahabad University, he went to my lab to set up the silicon facility for me. And he worked there for 6 to 7 years and got a number of publications with high citation. So that's very important that we set up the silicon facility in the faculty. So this is the research question. Can passion fruit extract reduce the risk for Alzheimer's disease? And with this pathway, the glutamate can induce cell death, particularly the neuronal cells. But in the presence of passion fruit, the cell number can be sustained, no problem with the high glutamate contents. So this is the picture. I tell you that the cell that we use called hippocampus cell, HT22. For example, the normal cell, they can survive very well, but with the glutamate incubation, glutamate can kill cell 50%. But in the presence of glutamate and the extract from passion fruit, the cell can still live it very fine. They can survive anyway. So from this picture, you can see that passion fruit extract prevent the cell death of glutamate. And then, in this picture, I try to show you that the cell death because of the high reactive oxygen species. The glutamate induce reactive oxygen species, but we can reduce reactive oxygen species to normal level because of incubation with the passion fruit extract, as you can see. And then I study in silicon. Here, basically, this normal silicon, they have 6 to 7 neurons, glutamatergic neurons. But one that becomes sick. So I mean, just 2 months ago, we published this. After 5 years, we found that the mitochondrial guardian, alpha myelin only, not beta, can help to work in the tau level pathway. So we found one of them that work very well. So this is very big paper, big publication, because the impact factor of this paper is over 14. So this is really big and very hard to publish in the level of impact factor 14. And we use AI to study. We gave the people to eat colorful fruits for over 10 years. Over 10 years, we have to follow the person who ate colorful fruit and who did not consume the colorful fruits. And we see the differences. And based on AI data, we have to find the interaction of the potential candidates with alpha or beta amyloid. And then we found only alpha amyloid that work very well. And also it can cross the blood-brain barrier as well. So using AI and the knowledge in pharmaceutical science, we can come up with this publication. Very important. And 2 actions that alpha amyloid did was, number 1, it inhibits the tau aggregation. Number 2, it promotes tau degradation. So this is very important to work to act in this pathway. So we know alpha amyloid action, and we know the pathway very well. And with this,
Sir, sorry to interrupt you.
okay,
Almost wrap up this session
okay,
within 3
sure,
minutes.
sure. I have one slide or 2 slides ago. And with this, we have to really prove the concept. And we got successful story because of the collaboration with many people, as you can see in this picture, worldwide actually, including Cambridge, Oxford University, and Norway also. So from one passion fruit down to 2 compounds and 1 single compound. In the future, we may develop this alpha amylin to be the drug. But now I can tell you that if you can try to eat more passion fruit, that may be a good way to prevent Alzheimer's disease. And this is my group in Thailand. We have a very big group with students, graduate students, undergraduate students, and we have five to six postdocs from India as well. So they came there to Thailand to work with me. And another thing, this is the last slide. Next month, in May 14, May 15, we will organize the conference in Bangkok, which is about AI and aging. So if you are interested to join, I would like to welcome you. Thank you so much for your kind attention.
Pleasure, Dr. Gavin, to get be the part of your session. Dr. Gavin not only make us aware regarding the problems associated with general public health, but also shed some light on the importance of natural products and its application to combat the various and distinct diseases. As his third area of research was neurological system and diseases associated with the neurosystem, he also embraced us with his lab work. Most importantly, Dr. Gavin highlighted that innovation is much needed in this modern era. Thank you, sir. Thank you for your words of wisdom. Now, I would like to invite sir for the felicitation.
Request Dr. Chitra from Amplify State, our speaker, Professor Dr. Devin. Thank you, sir. We now move ahead to the second technical session of the day. This segment continues our pursuit of scientific excellence, bringing new perspective and innovative insights from the field. It's my privilege to announce that we have Dr. Shikha Sharma as session chair for this segment as well. Ma'am, I request you to please take charge of the session and introduce our next plenary speaker, Dr. Chitra Arvind.
Welcome, Dr. Chitra Arvind, founder partner, Acquires IP Care LLP. Ma'am's qualification is PhD in Pharmacy, MBA, PG, DIPR, and LLP Advocate, Registered Patent Agent. Chitra Arvind is a founder partner in Acquires IP Care LLP, is a firm specializing in intellectual property rights in India. Earlier, she was an equity partner at Lex IP Care LLP. Acquires legal joint hands with Lex IP Care to form Acquires IP Care. She has a doctorate in pharmaceutical sciences, a postgraduate in business administration, specializing in marketing, a patent agent and a lawyer registered in the Bar Council of India. She has more than 2 decades of experience in prosecution of patent applications, drafting and filling more than 1,000 applications, and has handled several oppositions and litigations in various facets of IPR, specializing in life sciences and pharmaceuticals. She regularly conducts IPR workshops and is a visiting professor in many institutions across the country. Her multifaceted experience helps her in advising clients in building IPR portfolio and devising marketing strategies. She is an active member of international associations such as INTA, APPA, and AIPPI. I welcome you, ma'am, on the stage, and Please start your session.
Respected dignitaries on the dais, boys and girls, ladies and gentlemen, let's pep up. Okay? So, I was talking to Dr. Hema this morning. Okay, and all of you are Gen Z. Correct? Yes, ma'am. I have a teenage daughter, she calls me Gen Dino. Okay, so when a Gen Dino is talking to Gen Z, it's very difficult, right? Right? No? No? You people know AI better than us. Okay? Every time my phone hangs, I have to plead my daughter, yaar, thoda sa sahi kar de. Hai na? You do it for your parents? Okay? So why do you think that we should have a lecture like this? Just to tell you what to look, where to look. And I'm sure your chat GPT will give better answers than the lecturer. Right? How many of you are here in pharmacy or allied sciences because you failed in NEET? Almost everybody, I think. Come on, be honest. हम लोग भी बचपन में यही करके आए. But why do you think that you have landed up sometimes in luck in a better position than in NEET? 5 years MBBS, 3 years PG, assuming you get PG, plus 2 years after that, 11 years before you see money, correct? Here you will see money. How many of you wanted to be a doctor to be very, very serving to the society? Come on here, touch your heart and tell the truth. Okay, I believe you. You must be the one of. Okay. All of us want to make money. Why? Money runs the world. Correct? As long as it is ethical, principled, why not? If it is a product of your intellectual property and your brains, why not? Okay? So that is what we are going to see today. Bench to market. How do you take your intellectual property rights, which is a product of your ideas, to the market and monetize it? Okay? Okay, so we move to the first slide. I would like to have the first slide. Okay. How many of you know Benjamin Franklin? Yes? What did Benjamin Franklin demonstrate? Benjamin Franklin was a person who flew a kite with a key. Okay? He was US, one of the founding fathers, and he wrote the US Constitution. He demonstrated static electricity. Okay? Faraday's law, how many of us remember? Michael Faraday. Yes? Faraday's law. नीट करते वक्त हमने फिजिक्स पढ़ा था ना, इधर हाथ रख के इधर ऐसे मैग्नेटिज्म आएगा, ऐसे इलेक्ट्रिसिटी आएगा, भूल गए ना हम लोग. But you know, Michael Faraday was the person who showed the Queen of England. लंदन जाते हो ना, पढ़ा तो होगा. You know Thames River? You know Earth has electromagnetism? demonstrate is that there are two, this hand, what was it? One was magnetism, one was electricity, third was? Correct. We still remember our need, no? Pain is very raw still, right? So in these three hands, he was the first person to demonstrate that an idea can have an effect. Okay? Benjamin Franklin is not remembered so much, but all of you remember Michael Faraday, right? The story of Michael Faraday. Michael Faraday was standing before the English Queen at that particular point of time. The English Queen asked, and he was first demonstrating the Faraday's law to the Queen. He told the Queen, see, this is electricity. So the Queen asked, what is the use of electricity? Okay? You know what did he answer? What is the use of a newborn baby? A baby can grow into anything, right? But can you determine the use of the baby on day one? At that point in UK, there was a prime minister known as Gladstone. He said, you know, is this the way to answer to a queen? How can you speak like this? You know what Faraday said to Michael Gladstone? He said, sir, I agree she is the queen. I cannot answer her like this, but you, sir, are nothing but a prime minister, and I will answer you differently. He said, sir, electricity is something on which one day you will be able to levy taxes. Okay? What is this? This is bench to market. If your innovation is not from bench to market, what is the point of doing research? And that is sustainable research and development. All of us know that the funnel of pharma is very, very difficult. You get millions of drugs into one single drug. Okay? And all of you have ideas. But do you know that ideas are not protectable? So what is intellectual property rights? Intellectual property rights are protection of a tangible form of idea. Okay? When you convert your idea into patents, whenever you have scientific improvement, whenever you give a name or for anything, that gets protected under trademarks. The shape and configuration of any type of device gets a design protection. The expression of your idea is a copyright. Okay? Each one of these rights has its own law, own act, own judge, own office. Okay, so trademark, if you have a name, you cannot go and file it as a patent application. Understood? Okay. Now, within the patents also, it is possible for you to file it for a product like this, or you can file it for a process, or you can file it for the use, or you can file it for combination or compositions. What is the difference between combination and compositions? You're all suiting students, right? Right? Okay, non-suiting students, pharma students? Combination means two or more actives. Composition means two or more actives. on the shape and the configuration. For the technological improvement, I will have a patent. For the name of the product, I will have a trademark. And last but not the least, the software, okay, which determines the dose determination will be your copyright. Okay, all 4 forms of intellectual property rights. Okay, now why do you have all these 4 forms of intellectual property rights? You build a IPR portfolio. महाभारत पढ़ा सब लोग सोमत महाभारत पढ़ रहे हो पढ़ा है अभिमन्यु जानते हो यस अभिमन्यु का चक्रव्यूह जानते हो that's how your IP portfolio must be. Okay, nobody should enter your portfolio because it's a negative right. If somebody enters, they die there in litigation. Okay, so once you build a portfolio like this, then you go. When I go to most universities, people tell me, "Ma'am, we published in Nature, madam." Patent. Boss, publication and patents can go hand in hand, but the patent must be filed first. Okay? If it is your own publication, you still cannot patent it later. So always file the patent first, then you go and publish in Journal of Natural Products or Nature or Science. Not a problem, but the other way around is a problem. Okay, so you must always file the patent application first and then publish. Now, in filing a patent application, there are 2 types of patent application. The first patent application is a provisional application. Like in case of doctor's case, he has to work on an elephant. The elephant may be sleeping today. It may decide that I don't want to cooperate today. So research will take its own time. Your mice will die today. Okay. That is why you first file the provisional application. Then you get 12 months to file your complete application. From provisional to complete, you get 12 months. In these 12 months, you will complete the research, but you will never file for the complete application first. Okay. Now, when most of the NIRF rankings, I am telling this on a university platform, is meant to count the number of patents. Okay? Most of the NAC accreditation is meant to count the number of patents. Dr. Rao from BITS Pilani is very clear in the fact that patents should be marketable, and our new education policy bill, how many of you have seen that bill? Okay, there is
Sorry to disrupt you, ma'am. Please wrap up your session within 3 minutes.
Oh, ma'am, you have not realized what it is to give a lawyer to a mic. I will try to be as quick, but it is going to take more than 3 minutes.
It's also an immense pleasure to listen to you.
You give me a mic and no judge, so sorry. I will try to wrap up as quickly as possible, all jokes apart. Okay, now if you see patenting is step 1. When you tag the patent and convert it into the market and monetize, only that is most important. See, if you see the lifeline, you have to take it from the provisional application for 5 years till grant. Okay, if you have to take it for 5 years till grant, only then you can monetize your patent. Okay, now to see how your patent fits. If I go and ask people what you want to file, everybody says USA. How many cases of malaria are there in USA? See, doctor, if doctor had looked for Thailand Alzheimer's, why? Because of the aging population. You didn't think that doctor should work for Alzheimer's in a country with young population. We should be your decision drivers. Okay, so if you have to maximize your patent protection, you have to keep it evergreen. What is evergreen? What is the term of the patent? 20 years from the date of filing. But as pharmaceutical companies, doctor is not there. We keep it not 20 years. We keep it more than 40 years or 50 years. That is known as the gaming of patents. Okay, so if you have to do it, you have to do regulatory strategies, business strategies, drug discovery strategies, and patent strategies. To do all of this, because I have to wrap up the lecture, I will leave my presentation and go. You have to look at next generation products, starting from drugs, salts, ethers, esters, polymers. So takes children, okay? All of you must know, even if you are in allied sciences. Okay? And you have to do money matters. So that comes your pharma MBA as to how you will price the drug, where you will place the drug, how you will license the drug, and there has to also be mergers and acquisitions. Okay, you also have to look at how many of you do quality control. Is there a subject or paper of quality control? Yes? No? Maybe? Definitely? Definitely maybe? Okay, in the quality control, there is drugs where there are labeling instructions. How do you use labeling instructions? How do you use patent term extensions? All of you as fourth year students must have a quality paper. I will tell Dr. Nima and go. Wherever you are, in regulatory or in allied sciences. Okay? And you also have different kinds of patent strategies in which you start with a single drug and you go up to different kinds of drugs to formulate the chakra view of Abhimanyu. Okay, see, this is how many of us know adalimumab? Okay, this is Humira adalimumab. You have EPT people here, right? This is the structure or drugs of lenalidomide. Okay? And this is how omeprazole. Omeprazole we all know now. Antacids. That is the structure of omeprazole. Okay? So, last but not the least, this is the Benjamin Franklin we would like to see. You know what that is? A US dollar bill. Okay? All of you forgot the Benjamin Franklin in the US dollar bill. We start with Benjamin Franklin and end with Benjamin Franklin. But like this. Okay? I just managed to show you what you need to learn. Okay? You all are competing internationally, globally. Whatever we got, we got. Okay, out of need. But what we do out of this is what that makes the difference. Okay? So you can choose to be unhappy about your past, or you can choose to build your future. And if you want to build your future, the one and only thing that can build it is knowledge. Okay? We do not determine, I hire many people across many colleges. I don't determine where they are from. I determine what they know. Even if they don't know, we determine, do they know how to get it? And that's AI. Okay? a strong advocate for using ChatGPT in your work, but you should know how to use it. So use AI and monetize your intellectual properties to ensure that your ideas find real physical value in your bank. Wishing you all the best. Thank you, ma'am, and apologies.
It gave me an immense pleasure, Dr. Chitra ma'am, to be part of your session. The session was started with the importance of IPR as well as to empower the idea with the potential to monetize it. The session not only revolved around the concept of IPR but also shaped this session with your story, with the art of storytelling. Dr. Chitra also highlighted the 4 different types of pharmacy products designated as IPRs in the AI era. Thank you, ma'am. Thank you for the session.
Thank you, ma'am, for delivering a thought-provoking session. Your insights have certainly enriched our understanding. Now I request Dr. Shikha ma'am to felicitate our speaker, Dr. Chitra Arvind. Please give a round of applause. Thank you, ma'am. As a gesture of gratitude, I would now like to invite Dean SMAS to kindly felicitate our session chair, Dr. Shikha Sharma, for her valuable contribution in chairing the session. Along with Dean ma'am, I would like to request Ms. Kanishka Gupta, Associate Director, K.R. Mangalam University, to kindly felicitate our session chair. Ma'am, please. Now I request the SMAS to kindly present floral bouquet to our Honorable Associate Director, Ms. Kanishka Gupta, K.R. Mangalam University. Thank you, ma'am, for giving us your valuable time. We now move forward to the next technical session. This session continues our exploration of advanced research and technological innovation shaping the pharmaceutical sciences. We are honored to have this session chaired by Dr. Suman Saha, Associate Professor, Rajiv Academy of Pharmacy. I'd like to invite ma'am on the dais and invite our next plenary speaker, Dr. Rajneesh Kumar. Ma'am, please.
Hello, am I audible? A very good afternoon. I would like to invite Dr. Rajesh Kumar, an Associate Professor in the Department of Pharmaceutical Engineering and Technology at the Indian Institute of Technology, PHU, Varanasi. He is a recognized expert in analytical drug discovery with advanced training and notable achievements. In neurodegenerative disease therapeutics, particularly for Alzheimer and Parkinson's disease, he completed his postdoctoral training at the prestigious Karolinska Institute, focusing on the preclinical discovery of lead candidates for neurodegenerative disorders. Further, enriching his expertise, he conducted research at the Institute of Protein Research, Osaka University, where he worked on predictive modeling of ADMET parameters. With an extensive publication record and funding from both national and international agencies, Dr. Kumar's contributions have been recognized with Young Scientist Award of 2023 from the Bioinformatics and Drug Discovery Society, SRM Institute of Science and Technology, Tamil Nadu, and the BP Doctor Young Investigator Award presented at the International Symposium on Cholinergic Mechanism in France. Dr. Kumar has also served as an editorial board member for scientific reports, Nature Springer and Frontiers in Molecular Neuroscience. With this, I would like to invite him on the stage.
Okay, thank you so much for the kind introduction and to the organizers for the invitation. I'm pleased to be here. So I thought that I will deliver a kind of a research-based talk, but then I thought it might not be suitable for the young students. So I'll try to give a glimpse of. Not audible? Am I audible in the back till the end? No? Okay. So then I thought that, okay, let me give you a general introduction to what is AI and how we can use AI in drug discovery and development. Since I have only 30 minutes, if I'm not wrong, 30 minutes, right?
Yes, sir, sure.
Okay, so at 30 minutes I'll try to, sum up whatever we have been, doing in that area.
Sorry to interrupt, sir. You just have 2 minutes for that session. 20 minutes.
20 minutes. So this is a place where you can see like Nobel Forum, which is where the Nobel Prize in Vigilance in Medicine is being awarded, and that is done by Karolinska Institute Nobel Assembly of Professors. Okay, and I've been fortunate to work at Center for Alzheimer Research for almost 6 years, where we were working in the area of structural biology, neuroimaging, epidemiology, drug discovery, and even clinical trials as well. Coming to the main point, what is a drug? So if you look at the definition, so drug can be used to prevent, cure, okay, or treat the symptoms. So it can be prophylactic, which is preventing the disease. It can be therapeutic, which is curing the disease. It can be symptomatic, which is just dealing the symptoms with the symptoms, not treating the disease. Like we use paracetamol for treating the fever, which is not itself a disease, but it is just a symptom of the underlying disease, which can be anything from inflammation to cancer. Okay, so now and the drug discovery is a very challenging, split it into two parts: discovery and development, starting from the target identification to hit identification to lead validation, and then followed by ADME studies to become a drug candidate. So this whole pipeline is called as discovery, where you have to spend a lot of time, lot of money, and then the success rate is very low and challenges are very high. The same is with the development, where you start with a lead molecule, a drug candidate, and then you do clinical toxicology studies, and that is, for example, those are also called as IND enabling studies, where you have to screen those compounds, those lead candidates into at least 2 animal species, followed by clinical trials when those drug molecules or drug candidates are being tested in humans. Okay, after satisfying all these criteria, you get a molecule which now enters the clinic and which is available for being utilized by the used by the patients to cure the disease. And again, the challenge is that the time taken for discovering a drug from target identification to clinical trial is 10 to 12 years, and the money it is consuming is like around 2 billion USD. To give how challenging it is, I would like to share the story of omeprazole, which is a blockbuster drug. So, the company AstraZeneca, they started discovering this drug by identifying this compound, CMN. So this compound, when they identified, it was showing very good activity, but then it was shown to be, liver toxic, so they could not continue with this molecule. They have to identify another molecule, so they did that, and then they identified this another molecule which was having patentability issues because it was patented by some other company, so they could not go further with this molecule as well. Then they have to identify another compound which they did, but unfortunately that was showing again severe toxicity, followed by another molecule which was again showing severe toxicity, and finally after 7-8 years of intensive research they were able to identify this blockbuster drug which was named as omeprazole. Okay, so you can see how challenging it could be, the whole process of drug discovery and development, not only for us but for the big industries, right, which are having unlimited resources, unlimited funding and unlimited manpower. So then I have a question for you. So what will be the number after 8? All of you. Yes, what will be the number after 8? 10, right? How many of you agree with that? All of you? Yes? Right? So, yeah. But can you answer this question? Which of the following molecules are water soluble? Can you answer this question? Yes or no? Can you answer this question? Yes or no? Like
carboxylic acid. Which carboxylic acid?
So it's only carboxylic acid which makes water soluble molecules. So with the confidence we answer this question, are we able to answer this question with that confidence? Yes or no? No, right? Why? What is the reason why we are not able to answer this, but we did the previous one?
Multiple possibilities there.
Multiple possibilities are there, or I can say that we cannot identify the pattern. Can we say that? Here we can see the pattern, like every time we are adding 2 into the previous number to get the next number, we can identify the pattern, we can understand the pattern. But in this case, we are not able to identify the pattern, and that is where AI or computers or machine learning can help us. How they can help us? Because they are, fast, they can analyze large amount of data, the big data, huge amount of data, and also they work 24/7, right? And especially now when we have used computational power, even in our mobile phones, processors, they are much computationally powerful than the earlier Pentium computer. So this is, the overall, the arena of AI. So you have the big circle is AI, everything comes under AI, and then you have a smaller one which is machine learning, and then you have a smaller one which is representation learning, and then you have even smaller one which is the deep learning, and further you have transformer models and all. So and believe me, AI is in the air, because the time when you wake up to the time you go to sleep, we are using AI. Unknowingly or knowingly, we are using that. Okay. So in machine learning, what do we do is, it is called as predictive modeling. So in predictive modeling, what we do is we take the training data, we take the labeled data, and we use that labeled data to train a model, and then we use that model to predict the properties or predict the outcome of new molecules where we do not have that data associated with them. Okay. Coming to the main thing, which is how we can use AI in drug discovery and development. So as I said, drug discovery and development has several stages. and you can use AI in all of those stages. For example, you can use it for protein ligand interaction analysis, high throughput virtual screening, predictive toxicology, predicting admin properties, and biomarker discovery, or for designing the clinical trials. Okay. So like you can do the target identification. For example, a company called as In Silico Medicine. So what they did was they identified a target for this idiopathic pulmonary fibrosis, which is a deadly disease. So for this disease, they identified drug target, that is a TNI kinase, TNIC kinase. Okay. So what they did was they analyzed the multiomics data of patients as well as healthy volunteers, and from that multiomics data, they could identify the signatures which are responsible for the disease, and from those signatures, they could come up with the targets. So they identified 20 different targets, and out of those 20 different targets, they designed new molecules by using generative AI. By using generative AI, they generated new molecules, and then they could develop a molecule which is now in phase 2 clinical trials, and the name of molecule is Rentosertib. Okay. Likewise, you can do hit identification. For example, this is a tool called as deep docking, where you can screen ultra-large libraries like billions of compounds. If I want to screen billions of compounds, I can, with the help of this tool, I can do that. So that is, how you can use AI in hit identification as well. And then you have generative AI. So in generative AI, it is like there is, for example, how many of you are using ChatGPT or any other, okay, very nice. So if I ask ChatGPT, can you give me a list of journals who publishes drug discovery related papers? So it will give me a very nice list that, okay, drug discovery today, EGMC, GMC, science, okay, it will give me the list. But if I ask it, can you generate structures of molecules which are drug-like and which are, soluble, or which are permeable, or which are, like that, having specific properties. So it will not be able to generate those molecules. What is the reason? Can anyone tell me why it is not able to do so? Because it has not been trained on that kind of data, right? It knows English very well, it knows Hindi very well, it knows Spanish, Swedish, Japanese, German very well, because it has been trained on all those languages. It has been fed with billions of tokens, sentences, letters, words, but it cannot generate molecules, right? But there are tools which can generate molecules, which can give you the structure of molecules, and one of such tool is called as Reinvent 4. So in this Reinvent 4, you have multiple possibilities. You have the possibility, you have a generator model which generates the molecules, and then the generated molecules can be scored. They can be scored using scoring function, and this scoring function could be anything. It can be a QSAR property, or it could be a RTP descriptor. It could be a pharmacophore model. It could be a docking score, structure-based scoring, or it could be again a Campbell-based property prediction. Okay. So what it is doing is, now once it has generated a model, so then there are different types of machine learning model. One is called as supervised learning, another one is called as unsupervised learning, third one is reinforcement learning, fourth one is transfer learning. So do you know the difference between supervised and unsupervised learning? Can anyone tell me difference between supervised learning? What is supervised learning? No? Can you tell me? Yeah, this one.
Learning under someone's scale. Supervised learning.
Sorry?
Learning under someone who is skilled or knows about the subject.
Can you give me an example? Okay, I give you an example because we don't have much time. So, for example, I have a kid at home, 2 years old, and she has never seen a cat or a dog. Okay, we are walking outside in the playground, in the park, for example, and then we see a cat. Okay, now she's not knowing it is a cat, right? But then I tell her, okay, this is a cat. Means I am a supervisor. I'm telling her telling the kid that this is a cat. So now she will remember that it is a cat. Okay. Next day we are doing that again, and then we see a dog. Okay. And then I tell her that this is a dog. So now I have told her cat and dog. So under my supervision, she has learned what is a cat, what is a dog, what are those features. The next day, third day, when she will see a cat, so she will say it is a cat or it is a dog based on what it is. Okay, so that is supervised learning. On the other hand, if she is not knowing what is a cat, what is a dog, and then she see a herd of cats and dogs, like 50 cats, 50 dogs, she cannot say that this is cat, this is dog, but what she can do is, based on their features, she can segregate them. Okay, I can tell her that, okay, just segregate whatever you see based on whatever you observe. So what she can do is, because cats are smaller, have long, hair, fur, and everything, so she can separate, she can make a cluster of cats and she can make a cluster of dogs. So that is called as unsupervised learning. The third one is reinforcement learning. In reinforcement learning, that works on the principle of reward and punishment. Now on the third day, if she see a cat and then she say this is a cat, then she has made a correct prediction. So what I will do is I will give her a reward. For example, okay, tomorrow our playtime will be double. That is a reward. So she will be forced to do the correct prediction. Okay. On the other hand, if she see a cat and she say it is a dog, so then she made a mistake. So then I will tell her your punishment is that tomorrow will not go for play. Okay, you will have no playtime tomorrow because you made a wrong prediction. So that is called as punishment. So the same we do with the model. Okay, the agent is here a child and we are the supervisor. We are making him learn the patterns in the data, whatever kind of data we feed. It could be, the patent data, it could be the clinical trial data, it could be, formulation data, synthesis data, anything, any kind of data it can be. Now in this case we have different scoring functions, like you can have like molecular docking. The generator is generating a molecule. Now you feed this molecule to the docking method, for example autodocvina. Now this will generate a score, docking score. If the docking score is below cutoff, like minus 10 kcal per mole, then this score will give a negative or it will give a punishment to the model. So this generator will not generate molecules which are similar to that structure. So the model then will start exploring other molecules. Likewise, if the score is better, it will keep on generating, it will be a reward and it will keep on generating more molecules like that. Is that clear? So yeah, that is what is called as reinvent because it is a reinforcement learning based generative AI model developed by AstraZeneca. That's an amazing tool actually. You all can explore, and the best thing is these all are free of cost. Everything is open source. You can go to GitHub, you can go to read the code, run the code and learn. The next thing you can do is you can predict the toxicity. So next we can do the predict the toxicity. So there are several tools like DeepDD, Skin Doctor, DeepKit. So they can predict the toxicity of molecules. And basically these are QSR models have been trained on toxicity data. They can predict the toxicity of any molecule. Okay. Then you can do predictive ADMET, and I have been fortunate to work with Professor Mizuguchi at Osaka University, Japan. So what they have been doing is they are developing this tool called as DRUMAP, Drug Metabolism and Pharmacokinetic. So this is a very nice server where you can predict the properties, pharmacokinetic and toxicity properties of the molecule. So for example here in this paper, what they have done is they have predicted the fraction of unbound drug in the brain. Who is doing alpha pharmaceutics here? Alpha pharmaceutics कौन है? आप बताइए अनबाउंड ड्रग का क्या रोल है? What is the role of unbound drug?
It will bind to the protein.
Sorry?
It will bind to the protein, albumin and
Unbound drug. Unbound. Unbound. क्यों वहाँ देखिए लिखा हुआ है? Fraction of unbound drug.
it will bind to the receptor.
Very good. So in our body, thank you. So in our body, you have, when we take a drug, almost 90% of drug is bound to the plasma protein. It is not available, not free to go and, it is like, for example, you have, 1 CR in your bank account, but 99 lakh you have put in the, kind of PPF, which you cannot withdraw. You have that money, you can see, you can be happy, but you cannot withdraw. It's not liquid, right? The only which you can use is 1 lakh, right? So that is the same situation. 90% of drug is bound to the plasma protein. Only 1% is free, which can go and bind to the receptor and give the effect, right? So this is the relevance of this property, and they have developed this tool. And then finally, you can use AI in clinical trials, because if a drug is failing in phase 3 clinical trial, like when the company has invested billions of USDs in the project, if a drug is failing in phase 3 or phase 2, then the company is losing a lot of money, right? Isn't it? So it is better if we can use AI to design a better clinical trial, can reduce the risk of failure. That will be really good. So there is a tool called as PASAI-trials, which can be used to design better clinical trial by patient outcome prediction, by trial site selection, by doing the the patient trial matching, trial similarity, and synthetic data generation. Okay, but everything which is shiny is not gold, right? So we have some limitations, and believe me, there are a lot of limitations in the case of AI in drug discovery. So this was a recent news where the Sumitomo Pharma, they lost billions of USDs when their drug failed, which was developed using AI tech was failed in 2 phase 3 clinical trials. You can just imagine how much they have lost. And does anyone recognize this plot? It is called as Gartner hype cycle. Whenever a new technology comes, we believe that now it is done, it will solve all the problems. Okay? So, but it is not like that. We have a very high, inflated expectations, means it will solve everything, but soon we realize that it is not going to work, and then finally, after some time, we have the, the productive time. So, in the case of AI in image generation, text generation, music generation, video generation, we are already in the production stage, means we are already in the plateau productivity. But for AI in drug discovery and development, we are still, at the peak of inflated expectations. We believe that AI will solve all the issues of, medicine, and we will have a cure for all the diseases, but that is not going to work, believe me. So soon maybe we'll see, definitely see a trough of disillusionment, and finally maybe in a decade or 2 decades, we will have this, productivity. So if I talk about limitations, the limitations are unlimited. So the first thing is limited data. The data we have that is not reliable, that is not reproducible, especially the public data which is coming from publications, patents, and most of the data which is reliable is sitting with the companies, like big pharma companies. They have data, they are not happy to share. Okay. And another thing is complexity of biological system, because biology is so complex, you cannot, be 100% sure that it will going to work. Interpretability of models is another challenge, because the models, they look like black box in nature. We cannot say that, okay, this should work, but how it is working, when it is working, we don't know. Transferability of models is another problem. If I ask a very simple question, if I want to know the average height of Indian girls of age 20 to 25 years old, right? So that is my question. Indian girl, 20 to 25-year-old, average height. But I ask any of you to go to your village and take the average height of 50 girls and get the answer. Will that answer my question? Yes or no? No. Why? Did you get my question? I wanted to know the average height of Indian girl, 20 to 25-year-old, and ask one of you, like ask Garima to go to her neighborhood and then collect 50 girls' data and then give me that data. So will that data answer my original question? No. Why?
Sample size very small.
It's not about size actually. It's not about size. Because our sample is not correct.
It does not represent the actual population.
Exactly, because her neighborhood is not representing India, right? Yes. It is not that she is living in a mini India. So what we have to do is, every model has its own limited applicability. I cannot say that I trained a model of predicting solubility on 1000 molecules and that it can predict the solubility of 1 billion or trillion molecules. No, it's not going to work, right? Because that model is trained on limited data, so its application is only limited to that kind of data. So that is a very big challenge. Computational resources, we need ethical regulations, these are problems, collaboration, integration, cost and investment. And then, I think I will skip that. Already I've spent so much time, I think. But I can say, for example, we have been doing a lot of work on AI-based drug design. And this is, for example, we did for identifying cholinesterase ligands by screening 1.3 billion molecules. And then we were successful and we were happy actually. So in conclusion, AI is highly promising, okay? In accelerating drug discovery, it can improve the chances of success, it can increase the productivity and reduce the cost. However, there are so many challenges which are still, we are not able to solve, okay? And in the end, I have developed this 30 hours, 12 weeks course on AI in drug discovery and development, which is already live on NPTEL. So last year there were around 10,000 registrations. This year, July, it will be offered again. So those of you who are interested, they can register for this. And the best thing is that it is free of cost, okay? And you will not find this type of course anywhere else, because we worked very hard on it, developing this for last 5 years, okay? With those words, I would like to say thanks to the organizers and to the global audience who is listening to me for last half an hour. Thank you so much.
Thank you, sir, for your very, very insightful presentation on drug discovery and development using artificial intelligence. Your explanation of how artificial intelligence is accelerating target identification and lab optimization was more particularly impressive. And as far as we know that in this present landscape of drug discovery, we need to become aware of artificial intelligence, how artificial intelligence is transforming drug discovery and making the process more faster and precise. So in this present landscape of drug discovery, it's a biggest challenge how artificial intelligence is integrated with the real clinical data. So sir has explained us more a lot. Thank you, sir, for your insightful presentation. A big applause for sir.
Thank you, ma'am, for concluding the session in such a beautiful way. Now I request Dr. Sunam Saha to felicitate our speaker, Dr. Rajneesh Kumar. Give a round of applause. Thank you, sir. Thank you, ma'am. We now proceed to the fourth technical session of the day. This segment brings forward deeper insights into the environmental and ecological dimension of scientific research. I am honored to share that we have Dr. Sunam Saha as session chair in this segment as well. I request ma'am to kindly introduce our next plenary speaker, Dr. Rajesh K. Sharma. I invite sir to please raise the dais.
Dr. Sharma is currently an assistant professor stage, third at Department of Botany, Benares Hindu University, Varanasi, India, with strong interest in biomedical toxicology and specially focusing on their environmental monitoring, transfer and toxicity in plants, health risk and mitigation strategies. His Google Scholar Age Index is 25 with citation 6725. Dr. Sharma obtained his BSc, MSc and PhD degrees in botany from Benares Hindu University, Varanasi, India. Dr. Sharma has also served GB Pent, National Institute of Himalayan Environment, Almora for 7 years as a senior scientist. Dr. Sharma has executed research products funded by ENRF, DPT, GBPH, NIHE, UJC, IOA, BHU, and Sharma has published 2 edited books, 19 book chapters, 18 general articles, and 61 peer-reviewed research papers. Dr. Sharma has delivered keynote and invited talks and oral presentation in more than 40 national and international scientific meetings. Dr. Sharma has mentored 2 postdocs, 6 doctoral theses, and more than 40 master's dissertations in his research domain. His talk title is on contribution of cadmium-stressed medicinal plants in pharma industries. With this, I would like to invite him on the stage.
Thank you, madam, for your nice introduction. Good noon to everyone. First of all, I would like to thank the organizing committee for inviting me to give a talk in this conference. My talk is a little bit different from the talks which already given here, and the title of my talk is the contribution of the metal stressed medicinal plants in pharma industries. So how these medicinal plants, when exposed to the contaminated soil, cadmium contaminated soil, can be utilized in the pharmaceutical industries. You might have heard the name of this Franklin D. Roosevelt. He said that a nation that destroys its soil destroys itself, because whatever we are getting, that is directly or indirectly coming from the soil. Paracelsus, who is known as the father of modern toxicology, he said everything is medicine and everything is poison, only doses can make a difference. So it's the dose of the drug or anything which can decide whether it is a medicine or it is a poison. If we talk about the metal, these metals are coming from various sources and get accumulated in the soil, and the polluted soil is particularly affecting the ecosystem, and that influence is depend on the amount of metal present in the soil. According to the report of United Nations Environment Program, the 33% of the world agricultural soil are contaminated with the metal, and that is either moderate or severe contamination. In India, there are 330 sites which are probably contaminated with the metal, and most of the sites are located in the Uttar Pradesh. The areas like the Kolkata, UP, Tamil Nadu, Odisha, there are many industries which are emitting the metal that contaminate the soil, water, air, and also affect the public health of that area. These metals, when enter in the environment, affect the health of plant, affect the health of human. So they ultimately, if we are talking about the impact on the plant, so these are reducing the yield of the plant, particularly the agricultural crops, and the human being when exposed to these metals, either through inhalation or through irrigation or through dermal exposure, they are causing the toxicity in the human body. Here I have given you one example of the kidney failure. So this kidney failure is because of this metal. Basically this inhibit the enzymatic activities and that affect or that induce the oxidative stress in the cells that cause the kidney failure or the apoptosis of the renal cells. So what I want to say that these metals are affecting the plants as well as the human health. One example I am giving you here, that is the wastewater. The wastewater, either it is coming from the domestic area or from the industrial area. These are containing the metals based on the type of the industry, based on the types of activity in the domestic areas, and these wastewater is used for the irrigation purpose that enrich the soil with the metals, and these metals are entering into the food chain. Here the concentration of different metals is shown in the soil during 2007-2017 in the area of Varanasi and the Bhadoi. One study we did in the Varanasi, there is one river, Varuna river, and the water of that river is contaminated with the metal because many of the industries located near the that river, they are discharging their effluent in that river, and that river water is used for the irrigation purpose. So here I have taken the example of this rice crop. So this soil is contaminated with the metal, the water is itself contaminated with the metal, and different parts of the rice plant, whether it is root, shoot or grain, are contaminated with the different types of metal as I showed you in the figure. From market side, we have collected the 19 varieties of the rice. Say for example, Jeera Pattis, you know about this, Sonam, the Chintu, Basmati. These are the rice varieties that we were collected from the market side and analyzed the different metal in the grain part. And there are 6 varieties where the cadmium and chromium were detected. In other variety there the chromium and cadmium was not detected, whereas the copper and zinc were present. When we compared this cadmium concentration in this rice grain, their concentration was found above the permissible limit. So when we are consuming this rice, contaminated rice grain, so it will pose the health risk. Similarly, we have also collected different types of the vegetables, the grains from the wastewater irrigated areas. These are the the palak, radish, garlic, cabbage, this rice grain, wheat, brinjal, and we analyze different types of metal in these vegetables. And here I have shown you the cadmium concentration. This cadmium concentration was found above the permissible limit set by the Indian government as well as the WHO. Once the metal is coming in the environment and moving into different part of the environmental matrix, that is water, soil and the plant component. So we have to find out some solution, and the phytoremediation is the solution of this type of problem. In phytoremediation we can use the biological organism, maybe plant, maybe microorganism, or some soil amendments like the biochar, bio-oils, some fertilizers. So by using that, we can either extract the metal from the soil or we can stabilize the metal in the soil so that the metal is not coming in the edible part of the crop. Here I have done one study where I isolated 5 cadmium resistant bacteria from the wastewater irrigated soil and make a consortium of these 5 bacteria, and I found that this consortium is capable to increase the yield of the crop and reduce the metal concentration, particularly the cadmium concentration in the plant. So this is the related with the metal concentration in the environment, that is soil and the plant and the health risk associated with this contamination, and how we can remediate this soil contaminated with the matter. Now we can use the medicinal plant for the remediation. Why this medicinal and the aromatic plant are used for the remediation? If we are using the edible crop, here I have shown you, you can see in the below part of this diagram, here the food crop, that is the edible part, metal is coming in the edible part and we are consuming that food, contaminated food, so there is a health risk. But when we are using the medicinal plant, the medicinal plant will also accumulate the metal in its organ, that is root, shoot or leaf. But the main product of the medicinal plant, that is the essential oil or the methanol extract or the extract from the leaf or from the root, that can be directly used because the metals are not coming into the essential oil or the extract. So that can be directly used and there is no health risk associated with this. And the biomass of the medicinal plant can be used for some other purpose, say for example recovery of the metal, or the biomass can be used for the production of the energy. So I have surveyed the literature and I found that this the work on the medicinal plant is ongoing in different countries with the collaboration among the countries, and this collaboration is increasing, and there are one main area, that is the phytoremediation or study on the phytochemical profile of the medicinal plant, that is growing. And if we see the publication in this area, 57% of the published documents are the original research article, and some of them are the book chapter, like this 16% is the book chapter and 21% is the review paper. I have used 3 medicinal plants, and these 3 medicinal plants were grown in the cadmium contaminated soil, and we studied their physical, chemical and biological properties and the metabolite profile of these medicinal plants under the contaminated condition and non-contaminated condition. So these plants are Eclipta alba, Mentha arvensis, Plectanthrus ambonicus, that is Indian boras. So this is the experimental design. First we studied the cadmium accumulation in the soil. So what is the total concentration of the cadmium in the soil, and what are the concentration of the cadmium that can be taken up by the plant? So the concentration of cadmium is increasing in the soil with increasing the application dose, and similarly the accumulation in the the availability of the cadmium is also affected by the concentration. So here you can see the total cadmium in soil increases from control to T4 in all the plants, and Eclipta alba is accumulating the highest concentration. Okay, and if we see the residual concentration, it is very high in the control, but in the T4 the iron manganese oxide bound and carbonate bound concentration enhanced. Similarly, the metal concentration in the plant is also increasing with the increasing concentration, and these metals are accumulating in different parts of the cell, either it is cell wall or the cytoplasm or vacuole. So this concentration, this accumulation is also affected by the increasing concentration of the cadmium in the soil. If the accumulation in the plant is higher than the concentration in the soil, so that plant can be used for the phytoremediation. So these are the some response of the plant to the contaminated soil. That is the total plant biomass, total length at different age, that is vegetative age and the reproductive age. So these biomass and the total length of all the studied plant decreased significantly by the treatment of cadmium. Similarly, the physiological activities such as the transpiration, photosynthesis at different age of the plant were negatively affected, and that effect was significant. The lipid peroxidation, that is the damage of the membrane of the plant cell, were increased because of the cadmium concentration at both the age in all the three plants. The enzymatic activity is also increased because plant is capable to combat the negative effect of the cadmium. So for this, the enzymatic activity in the plant is increased under the cadmium stress. We have also studied the stomatal structure, the trichome density, the oil gland density. So there is significant negative effect on these parameters in these three plants. Here you can see the stomatal bed negatively affected, that is decreased. The stomatal length increased, stomatal density increased, and the trichome density also increased. This is the oil gland secretory gland density that increased in the presence of the metal. This is for the Ambonicus Indian borers. Here the stomatal density also increased, the secretory gland density also increased. So if we see the antimicrobial activity of the methanol extract, so here the inhibition zone is increased by the cadmium treatment. So we have used some microorganism, that is this Pseudomonas aeruginosa, Klebsiella, Salmonella, Enterococcus. So these are showing the positive effect on this antimicrobial activity. We have studied the total phenolic content, total flavonoid content in the stressed plant and unstressed plant. So because of the cadmium stress, these metabolites, these secondary metabolites, phenolics and the flavonoids increased in all these three plants. The antioxidant activities in terms of the DPPH, ABTS and the FRAD, these activities were also increased and significantly increased in these three plants when these plants are exposed to the metal stress condition. We have performed the GCMS profile of the essential oil, and there is significant change in the metabolite composition of the essential oil. So here you can see in case of this E alba, Eclipta alba, in control condition, the caryophyllene is the maximum concentration of the caryophyllene was observed, whereas in the cadmium treated plant, this production is decreased and the maximum production is observed for the isoflavone, that is 56.33%. So there is a significant change in the biosynthesis of the bioactive compounds. There is significant change in the eucalyptol. There is 228% increase in this compound due to the cadmium treatment, and caryophyllene oxide that decreased by 12%. And these two molecules are having different medicinal properties like the anti-inflammatory, bronchocoagulated, mucolyte, etc. This is for the mentha arvensis. Here the maximum production of the cerolone, that is 70% in the control plant, but the maximum production of the 3-octanol, that is 26.79%, is observed in the cadmium treated plant. There is also the significant change. There is an increase or decrease. So here the eucalyptol decreased by the 55% and alpha cardinal that increased by 36.4%. This is with the Ambonicus plant. Here in control, the maximum concentration of carbachol was observed, that is 80%, whereas in cadmium treated plant, the concentration of the orthothymol was observed maximum, that is the 50%. So there is significant change in the composition of the bioactive profile of the essential oil. Here beta pinene that decreased by the 60%, that is the maximum one, and cadena 1,10,4-dine which increased by 64% because of the exposure of the medicinal plant, this particular plant, to the cadmium contaminated soil. We have also performed the UHPLC HRMS profile of the methanol extract obtained from the cadmium treated and untreated medicinal plants. So this is the chromatogram of the three plants, that is the control and the treated plant. In case of these three plants, some compounds increase, some compound decrease, and some compound there is no change. So these upregulated metabolites, which likely belongs to the phenolic, flavonoids and the alkaloid classes, contributing to the antioxidant defense, and the downregulation of the compound that indicate the suppression of the primary metabolism under the cadmium stress, and the compounds which are not changed. So basically the common compound that share the cadmium induced defense mechanism in the plant. So the plant have to protect themselves from the cadmium contaminated soil. So some compounds are contributing in that aspect. We also perform one experiment by using this tulsi, two variety of the tulsi, tanniflorum and the basilicum, and these plants were exposed to the different concentration of the cadmium. So here you can see the cadmium accumulation in the root, stem and leaf is increasing with the increasing concentration, and the maximum accumulation was observed in the root, followed by the stem and least in the leaf for both the variety. We have observed there is significant increase in the production of the essential oil in the Osimum species because of the treatment, because of the cadmium treatment. So this cadmium treatment is can be used to enhance the oil production, to enhance the antimicrobial activity, to enhance the antioxidant activities, but there is one problem which is related with the accumulation of metal in the biomass. So we cannot use the biomass directly for the consumption purpose because of the high concentration of the metal, whereas we can use the essential oil as well as the extract from the cadmium treated medicinal plant for the pharmaceutical uses or for the pharmaceutical purpose or for the health benefits. So these medicinal plants can thrive on the metal contaminated soil. They produce the essential oil as the main product, which are safe for the industrial use. So we have to identify which molecule is increased, and we have to isolate that molecule. We have to study the toxicity of the that molecule or benefit of the that molecules, and the remaining part, the biomass which accumulate the metal concentration. So there is further need of the study whether the metal accumulated in the biomass can be is available for the absorption. The bioaccessibility of the metal should also be studied. It means the metal present in the biomass, if it is consumed, all the metal present in the biomass will be absorbed in the human body. This is my lab, and we have students, and these are the funding agencies that have supported our studies. So thank you very much for listening me. Thank you, ma'am.
Thank you, sir, for your very insightful presentation and contribution of cadmium-stressed medicinal plants in pharma industries. It was particularly fascinating of how cadmium-induced stress is generally considered harmful, but still it may enhance the production of secondary metabolites like flavonoids, alkaloids, phenolics that can turn the stress into an opportunity for the drug development. And he also explained that how controlled stress can be used as an eliciting strategy to improve the pharmacological bioactive compounds. That was really especially thought-provoking. And he also experimentally has shown that how we can balance the beneficial increase of bioactive compounds with the risk of cadmium toxicity in the medicinal plant-derived plants. Thank you, sir, for your section chair. It was an immense pleasure to listen. Thank you.
Thank you, ma'am, and heartfelt thanks to our speaker for sharing a knowledgeable and brainstorming session. Now I request Dr. Sunam Saha to felicitate our speaker, Dr. Rajesh K. Sharma, as a mark of appreciation. As a token of our respect, I now invite Dean SMAs to kindly felicitate our session chair, Dr. Sunam Saha, for her valuable contribution. We now move ahead to the fifth technical session. Continuing the momentum of knowledge exchange, this session focuses on industry perspective and quality assurance in pharmaceutical sciences. We are honored to have this session chaired by Dr. Chandra Mohan, Associate Professor, School of Basic and Applied Sciences, K.R. Mangalam University. Sir, please come to the dais. I request sir to introduce our next plenary speaker, Dr. Sandeep Sutriyal, and welcome him on the dais.
Thank you for the introduction. So, moving ahead, our next speaker is Dr. Sandeep Sutriyal. He is an associate professor in the Department of Pharmacy, West Kalani campus. Dr. Sundial has earned his PhD in medicinal chemistry from NIPAD, Mohali, in year 2008, and between 2009 to 17, he worked as a postdoc fellow at various international institutions, including Baylor College of Medicine, Texas, A&M Health Science Center, and Imperial College, London. Dr. Sundial's research focused on design and synthesis of novel antimicrobial, anti-proselytic, and anti-cancer agents. 3 of his PhD students have graduated, and currently he is supervising 5 PhD students with projects addressing drug delivery, drug discovery against various protein targets. His group has worked extensively for the design of new anti-malarial molecules and recently characterized an anti-malarial chemical space. global recognition such as Harry Asia Outstanding Thesis Award and Marie Curie Incoming Fellowship. He has published over 45 research publications and review papers in independent journals. He is a life member of Chemical Research Society of India, PASAI, and a fellow of higher education in the UK. He continues to contribute actively for pharmaceutical research and education. So we welcome Dr. Sundayal. He will speak on harnessing anti-malarial chemical space and machine learning for the discovery of novel anti-plasmodials. Welcome, sir.
Thank you. Thank you for the kind introduction. So I would like to thank all organizers for inviting me here and all the speakers for enlightening us in this conference. So you have heard a lot of talks related to AI. Now I'm sure most of you have a pretty good idea about AI, machine learning and their applications. So in my work, we use cheminformatics and machine learning in a particular area to identify new anti-malaria. Now I'm sure students know about malaria. Can anyone tell me which organism causes malaria? Mosquito is a vector which spreads malaria. So which is the parasite? Excellent. So Plasmodium, it has 4 species and mostly Plasmodium falciparum is the causative organism, right? So we are looking for molecules which can kill this parasite, and we use cheminformatics. And this slide about drug discovery, already Dr. Rajneesh showed you that drug discovery is a long process. It consumes a lot of money. So we want to reduce this process because we want to reduce the cost, and especially for treating malaria. I'm sure you know which region of the world malaria is mostly, we observe this malarial infections. Which countries? Anyone? Africa. Yes. So African countries, their population suffers by malaria a lot, and you know it is low-income country, so they cannot afford very expensive drug. So obviously we have to decrease the amount of money involved in discovery of new anti-malarials, and that's where artificial intelligence and machine learning tools can help us. So now how to find a drug? Because if you see overall possibility of drug, there are actually you cannot count it, but one estimate is that there are 10⁻60 molecules possible theoretically. If you combine take all possible combinations of carbon, hydrogen, nitrogen, all these basic elements, you can make a lot of molecules. So now there is this space is very huge. How to find drug-like molecules? So anyone from medicinal chemistry who can define drug-likeness? How do we define drug-like compounds? Any rule they have studied? Lipinski's rule of 5. Yes, excellent. So Lipinski's rule of 5 was given by Lipinski, and he gave certain criteria for molecular weight, for the partition coefficient, which represents the lipophilicity of the molecule, then its hydrogen bonding capacity. So he gave some rules which are defined, or they are named as Lipinski's rule of 5, and they say that the probability of permeability of such molecules and solubility increases if they follow these rules. Now similarly, within these broad drug-like molecules, for the particular target also there are obviously some properties of the molecule. For example, molecules which inhibit kinases, they will have certain properties, proteases. Drugs which can pass the blood-brain barrier, they will have some properties, right? So within broad drug-like space, there is individual species, and that is because each target have their own binding pocket which defines the type of molecule it can interact with. Right? So location of the target or its size of the binding pocket or nature of the binding pocket will determine what type of molecule can bind it. Now malarial parasite is interesting in this way because you know malaria, asexual stage of malaria is within the red blood cells. Right, so red blood cells, if it has to, because if it has a molecule has to kill the parasite, it has to cross, first of all it has to go to the blood, then it has to cross the membrane of the red blood cells. And then after that there are two other membranes of the parasite. And these membranes are special because they have permeability. The permeability is very, very different because the parasite can induce new permeability pathways here. So at least 3 membranes it has to cross, any molecule. So we thought that because of these restriction or these membranes, special property of the membranes, can we have anti-malarial likeness or anti-malarial like compounds? Because are these membranes or the location of the parasite giving a special, only making special molecules to act as like an anti-malarial molecule? So we collected some data from the literature. So we divided them into research antimalarials, advanced stage antimalarials, that means antimalarial drugs or which are in clinical trials. And within the research stage antiplasmodial molecules, also we had low active, moderately active and very high active molecules. So 3 classes. So we named as HA, MA and then inactive cells. And we compared their properties with drugs, oral drugs, other oral drugs, and we studied the same properties, molecular weight, lipophilicity, C-log-P, number of aromatic rings, TPSA, which is again polar surface area. So in all cases you see we found some pattern that molecular weight increases if the activity or if the potency of malaria-induced molecules increases against the parasite. Similarly, they are more lipophilic compared to oral drugs. That means antimalarials. Antimalarials are more aromatic. They have more aromatic rings, but their TPSA is less. That means their polar surface area is lower compared to oral drugs. So this was some pattern. In addition, we find that basic nitrogen is a very important property because although nobody has studied it yet in case of malaria. Because if you see the structure of all the drugs in the clinic as well as in clinical trial, they all have at least one basic nitrogen, almost all. There is obviously some exception, but most of them have the basic nitrogen. So we studied this property also, and indeed we found advanced stage malarials and highly active molecules, antimalarials, have more, on average, more than one basic nitrogen compared to oral drugs. Then we studied the high throughput screening data. So in high throughput screening, we take a big library of molecules and screen against our target. So in this case, the target was different stages of the parasite. So there are mainly 3 stages of parasite: asexual, gametocyte and liver stage. And we did the same study, and we found that again the hits which we get in this, that is active molecules in this assay, again they are more basic. In addition to being more basic, again they were more lipophilic, they were more bulky, that is bigger size and less polar. So that was interesting, and we showed that the hit rate, if you see, of this library, original hit rate is 0.53. Okay, so that means you can see library size was around 68,000 molecules, only 363 molecules were active after what they found. But suppose if you remove all the basic nitrogen containing molecule, if you remove, hit rate will fall, which again shows that hits are more basic basically. So if you put a criteria that only we will take basic molecules in the library, that is basic nitrogen at least 1, then hit rate is 0.92, almost double. And if you put the criteria of 2 basic nitrogen, hit rate further increases, doubles almost. So that shows that again, if you pre-screen the library before doing high throughput screening, you can increase the hit rate, and that means you do not have to randomly select the molecule for your experimental studies. you can put a criteria of basic nitrogen. Similarly, in addition to basic nitrogen, if you combine C-log P, TPSA and aromatic ring criteria also, you can further increase the hit rate. And that was that led to our overall, you can say it is kind of a rule we have prescribed, which we call anti-malarial chemical space, and these are the filters we have given, that basic nitrogen should be at least 1 or more than 1, C-log P more than or equal to 2, aromatic rings more than or equal to 2, and TPSA less than 80. So if we pre-screen our libraries with these criteria, we have more probability of finding anti-malarial molecule in the experiment. So this can reduce the overall time of the study obviously, and also the cost. But if you see the commercially available libraries which are available to purchase and which are generally used for high throughput screening studies, these libraries do not comply to this chemical space. So we found that commercially available libraries are not very suitable for discovering new antimalarials because they do not follow these filters. So that means, so again AI can help here, because as Dr. Rajneesh told us in the morning, we can use generative AI methods to design new molecules which are not available but which can be synthesized. So that's what, that's where we will have to go now. Then there is another study we published recently, which is related to machine learning model. So here we collected, again we have in the literature, we have a lot of data of the antimalarial molecules. So based on the IC50 values, we classified molecules. So it was like a two-class problem here. So either we have inactive molecules or active molecules. So that kind of data we fed to the machine learning model and trained it. So this is how we divided data into training set and test set. And then, as Dr. Rajneesh told us in the morning, there are different types of models, algorithms. So out of these, we selected random forest.
Participant, please pay attention to the speaker. Please.
So random forest model is usually a very good, very fast, as well as it is resistant to overfitting. There is no overfitting here. So we found also there were many applications in medicinal chemistry for this. So we use random forest to develop the model, and then there are different validation techniques, because after you generate a model, you have to validate it using obviously the training set data, and there are other methods. So we found that it is a good model, robust model. And again we use different fingerprints, because here we have to train the model based on certain properties of the molecule. Here we selected the structural feature, which we call fingerprints. So out of 10 types of fingerprints we tried, and this Avalon fingerprint gave us the best result in terms of accuracy. And also we calculated the applicability domain, because as Dr. Rajneesh told us, it is very important that we know the limitation of our model. So what type of molecules you can predict with high reliability. So that is important. So that also we confirmed. And then we screened through these models a library of molecules which are already in clinical trials, because we wanted to see the repurposing of this molecule, whether it is possible or not. And we found several hits. Out of these hits we purchased these 5 molecules, 6 molecules, and you can see out of these 3, these 2 are the most potent molecules, which showed activity against the parasite. It killed the parasite, and later we also found that it is inhibiting the beta hemetin. So if you remember, one of the target is for antimalarials is hemozoin synthesis. So same hemozoin synthesis we can reproduce in the lab in vitro, and we call it beta hemetin. So if these 2 molecules were found to inhibit that beta hemetin, which we propose as their main mechanism of action. And these 2 drugs are both kinase inhibitors. So they also show the antimalarial activity. So overall, it shows the application of this machine learning model, and you can reduce the time. So that is the most important thing, because the time is money, we all know. So overall process of finding hit and then converting it into lead will reduce. That will reduce the cost. So that was the main aim. So with this application, we propose that we can use now the combination of these our rules, filters, anti-malarial chemical space, as well as machine learning models in combination to quickly screen large libraries, which may be commercial libraries or generated through de novo methods, and that can reduce the overall time and cost of making new antimalarials. So this is some of the publications from this work. So I have tried to kept it very succinct, because most of the details I cannot give you here, technical details. You can find it out. But overall application, I have showed you how chemoinformatics and machine learning combination can quickly give us antimalarial leads. So this is my PhD students, our group, and I would like to acknowledge my collaborators at IBAB, Dr. Shruti, Professor Gopal from ISC Bangalore, and Dr. Pooran Sinjwani from CCMB. Also, I would like to thank ICMR, SERB, and DBT for sponsoring some of this work. So finally, thank you all, and I would like to invite you to our West Milani campus whenever you get a project. Thank you.
Any questions from the participants? Questions and queries are open. Please ask. Yes, anyone?
I think we are close to lunch, so everyone is hungry.
Maybe.
Do anyone have any questions? This side?
Sir, I have a question. As you discussed, AI and machine learning are very important to identify such kind of plasmodial drugs for the health disease, to prevent the health disease. So, is it useful for the fathers, to assess or to predetermine the kind of disease we are having, type of disease? Other yeah.
Yeah, so these are just the tools. You can apply them for any other target. All you need is just good quality data. So that is the most important thing. So if you, for any other target or disease, if you have good quality data, then you can train model on that data, and obviously you can get the solution.
Any more questions? So, we want to thank you, Dr. Sandeep. It's a privilege to listen to your speech and your lecture, who delivered a session on anti-malarial compounds and machine learning for anti-plasmodial activity. This topic lies at the critical intersection of medicinal chemistry, computational science, and health issues. Malaria caused due to Plasmodium parasite pose a significant resistance and require urgent attention towards effective anti-plasmodial agents. So, Dr. Sundar have introduced about the paradigm shift for chemical space to explore unidentified compounds for therapeutic potential. Machine learning and AI can play a crucial role in transforming immense chemical space. So, we are fortunate to have a esteemed speaker. So, thank you once again for introducing such an important topic in present day. Thank you, sir.
Thank you, sir. Now I would like to request Dr. Chandra Mohan to felicitate our speaker, Dr. Sandeep Sundrayal. Thank you, sir. Now moving on to our sixth technical session of the day, I am privileged to announce that Dr. Chandra Mohan, Associate Professor, SBAS, KR University, will remain session chair for this segment as well. Our next speaker is Dr. Sandeep Sharma. I would like to invite him on the dais. Please put your hands together for him.
Thank you, ma'am. So, I invite Dr. Sandeep Sharma, who is a pharma PhD, and he is a executive quality assurance at Torrent Pharmaceutical Limited. He brings over 11 years of specialized experience in pharmaceutical quality assurance and management system within a European United Certified Regulatory Plant. He is known for his expertise in operational excellence, regulatory compliances, and strategic implementation of robust quality management system. Aligned with current global standard and with deep technical proficiency in process validation, particularly cleaning validation for effective cross-contamination control, along with extensive experience of managing CAPA systems and resolving market complaints to drive continuous quality improvements. His doctoral research in radio decontamination using animal models and regulatory aspects of radio pharmaceuticals further strengthened his scientific foundation. In his expert talk on pharma and allied sciences in the age of AI, innovation and IPR strategies, he will be sharing valuable insight on the application of artificial intelligence for manufacturing excellence and enhanced productivity. With this, I would like to welcome and invite him on the stage.
A very good afternoon to all the industry leaders, respected faculty members, students, and dear colleagues. First and foremost, I would like to thank the staff and the organizing committee of KR Mangalam University for having me here. When I was called upon by one of the faculty from KR Mangalam University to deliver a talk on AI in pharma, one thing was very clear in my mind. I wanted to make a presentation which is as simple as it could be, because my end audience are the students. Just like AI is the future of pharma industry, you students are the future of pharma industry as well. So I would like that you get acclimatized to all the intricacies of AI so that you can do it in the practical way when you finally reach to the pharmaceutical industry. My talk will be on navigating the intricacies of pharma industry using artificial intelligence, prospects and perspective. Starting with a very brief introduction and explanation on artificial intelligence. AI is the field of computer science that develops systems capable of performing tasks traditionally requiring human intelligence, learning, reasoning, decision making at scale. In pharma specifically, AI means deploying machine learning and deep learning algorithms to ingest massive amount of data from production lines and execute tasks that once required direct human intervention, making it faster, more accurate and continuous. So, it is important to understand what are the current challenges which the pharmaceutical industry right now have. First and foremost is the machine breakdown. The pharma industry operates on 24/7 basis, and it is very important that all the machines they run on 24/7 basis, because the unplanned downtime disrupts production, schedules and increases cost. Another point to ponder upon is the deviations. The quality incident triggers lengthy investigations and compliance risks. The market complaints the product defects damage the brand reputation and patient trust. The supply chain disruption. These days you are saying that there is a massive supply chain disruption. Geopolitical and logistical volatility threatens medicine availability and cleaning validation. The complex protocols consume time and resource across changeovers. So AI has the answer to each of these problems. Let me take it one by one. The vibration fingerprint approach. You must have noticed a train moving through the bridge. When the train moves through the bridge, there is a particular harmonic sound which emerges from this, right? In the same way, when the machine operates, just like a compression machine operates, it has a particular sound which continuously emits from it. We teach that sound of a healthy machine to the system, and whenever there is a likely breakdown to occur, the likely breakdown to occur, the AI models generate a trigger to the operator and it tells them that the machine is not operating on the required basis. Deep learning models, they are trained on acoustics and vibration fingerprints of the machines in normal operation, learning to recognize the signature of healthy equipment, right? How it works, the AI detects analogous harmonic signatures, subtle sounds and vibrations that typically emerges 2 to 3 weeks before the machine breakdown really occurs. So we go from the reactive repairs to a proactive maintenance, because in the pharma industry we have a particular schedule of preventive maintenance. All the primary equipments, their preventive maintenance is done on a 6-monthly basis. The secondary equipments, the preventive maintenance are done on yearly basis, and likewise for the ancillary equipment. So the timely detection of a machine breakdown can be done through the AI. The failures identified weeks in advance, dramatically reducing the unplanned downtime events. 90% confidence threshold. Maintenance is scheduled precisely when the AI predicts a 90% probability of imminent failures. And proactive shifts, operation moves from reactive repairs, just like I was telling, we have a planned schedule. Now, these days we don't go for the planned schedules, but when the AI sensor detects there is a machine, likely breakdown of machine to occur, so all the preventive maintenance activity comes then and there only. AI in QMS, reducing deviation with natural language processing. An LLM engine is integrated directly into the QMS of the system, creating an intelligence always on deviation response layer. How it works? When a deviation occurs, the AI analyzes the live flow data and compares it with 10 years of historical data, reports and kappas, instantly surfacing the most probable root cause. The results, we have a validated kappas with us, and all the deviations can be sorted within few hours, you can see. The market complaints, the computer vision. You just see this. This is a conveyor belt from the primary machines, from the primary cubicle. These bottles are coming, right? And you can see a circle. In these circles, we have the AI cameras. What we do is, in all these cameras, we teach the model that this is a perfect case of a bottle coming from the primary packing line, and the AI detects. We have a gold standard for this. We give the AI the command that this is a perfect case of bottle which is coming from the primary source. And when it comes to a secondary line, if there is any, you can say, a problem at the sealing level or problem with the printing level, the AI detects it, and automatically these bottles, they get rejected and are not packed into the final packing containers. The AI intervention, high-speed convolution neural are installed at the end of packing lines, performing real-time visual inspection on every unit produced. How it works? The cameras capture 360-degree high-resolution images of every product details. The AI is trained on synthetic data covering every conceivable defects like cracked glass, loose screens, and smudged labels. So, we have the zero market complaints, catching the invisible. The market complaints related to physical defects, they are driven to zero through continuous AI-powered inspections. Since I told you that we have a master, the gold standard is already put into the machine, and the machine
on a 360-degree basis, it continuously detects what are the possible defects in the machine, or possible defects in our final product. Beyond human vision, the system detects micro cracks and defects previously invisible to the traditional automated inspection methods. High speed, full 360-degree coverage maintained at high throughput, no speed or accuracy trade-off required. Resilience in global supply chains. Supply chain disruptions is not merely a business hurdle, it is a patient safety crisis. The AI control towers now deliver end-to-end visibility to navigate geopolitical and logistic volatility in real time. Demand forecasting by integrating disease surveillance data, weather patterns and social media sentiments, AI reduces the forecast errors by 20 to 50%. End-to-end visibility, AI control towers monitors the entire supply networks, enabling proactive response to disruptions before they impact the final patients. AI integration in cleaning validation. Tablet manufacturing faces persistent problems. Just in our company also we have almost 200 different molecules, and all these molecules they are having different solubility and they have different partition coefficients, right? So how AI can help us in this way? Because our cleaning agent always remain the same. This is the water, and at the most we have the 0.25% weight by volume SLS, sodium lauryl sulfate. The cleaning agent remains the same, and how we can integrate AI in cleaning validation, I will right now tell you. The tablet manufacturing faces persistent challenges like sticky granulations, highly potent APIs, multi-product equipment sharing. AI is transforming how we validate, monitor, and optimize cleaning process across life cycle. Predictive cleanability modeling. Before a swab is taken, AI ranks the worst case product, replacing trial and error with molecular analysis. The AI algorithms analyzes chemical structures just like the researcher was telling. AI algorithm analyzes chemical structures, solubility and surface adhesion of a new API to predict residue behavior. Surface interaction, because in all the equipments the surface remains the same, 360 stainless steel, right? To surface interaction. Simulating how tablet formulation interacts with 316 stainless steel or polymer seals to identify the hardest to cover residues. Worst case ranking. AI generates a scientifically defensible product case ranking for validation protocols, eliminating months of empirical testing. The intelligent visual inspection. In the pharma industry, we have production people, we have quality assurance people. Whenever there is a machine changeover, line clearance is offered to the quality assurance personnel, and he offers the line clearance by visually checking the machine for its cleanliness. For me, the degree of cleanliness can be different. For someone else, the degree of cleanliness can be different. So how AI can be used in this way? The intelligent visual inspection. The traditional visual criteria is subjected. The traditional visual criteria is subjective and error prone. AI-powered computer system replaces human judgment with quantifiable, repeatable detection. Multispectral imaging. High-resolution cameras with UV and NIR lights, they are trained to recognize the spectral fingerprints of specific recipients or API or equipment surfaces. The automated residue detection. AI detects residues at micro levels invisible to the human eyes, flagging hotspots on the tablet press turrets or CFD spray walls with zero subjectivity. Smart clean in place optimization. The traditional CFD cycles are over-engineered. You must be having washing machines in your home. There are different modes of operating washing machine. There is a simple mode, there is a full mode, there is a mix mode, right? Whenever you operate it, there is a particular recipe and the system, and the clothes are washed according to that recipe. 45 minute cycle, 30 minute cycle. But sometimes what happens is the clothes, they are actually not clean. So how AI can be integrated into the system for the machine, for the pharma industry, just like auto coaters. I will give the example of auto coaters. Traditionally, CIP process cycles are over-engineered, excess water and detergents just to be safe. AI-driven technology optimizes every cycle in real time. The sensor integration. Real-time conductivity, total organic content, flow rate, temperature and pressure data, they are fed into the AI model continuously. Dynamic cycle adjustments.
AI analyzes the TOC decay curve and terminates the cycle the moment residue is confirmed gone, saving time and resources, because the regulatory is very strict on usage of water even, because we have to optimize the use of water also. Anomaly detection. Flow turbulence analysis identifies shadow zones where cleaning fluids is ineffective, preventing validation failures before they occur. AI and operational excellence across unit operations. AI integration extends beyond cleaning validation. It delivers measurable impact across every unit operation in tablet manufacturing, from granulation to compression and coating. Granulation, the NIR-driven endpoints detection for precise moisture and particle size control. In drying, the real-time moisture profiling prevents over or under drying. In compression, the predictive weight and hardness control, it reduces the rejects. And in coating, atomization, air pressure control, and preventing overwetting of the tablets. Let us move on to AI integration in granulation. How many of you have gone for the pharma industry training after third year? Anyone? Anyone had seen the pharma industry at the core level? Any student? I am particularly interested in the students. There must be fourth year students.
4 weeks.
There must be
4 weeks industrial training is mandatory as per PCI syllabus.
How many of you have gone for the industrial training?
4 week industrial.
You must have seen the fluorides, FPDs. Any one of you have seen the FPDs? Right? So how AI integration can be used? What we do in pharmaceutical industry is, after granulation, after the RMG process, the system is fed into the FPD, fluidized bed dry. Due to the automation air pressure, all the granules are there fluidized, and the endpoint detection of that all the fluidization process is the LOD achievement, loss of drying achievement, right? How AI integration can be used in the LOD detection? For LOD detection, the operator has to stop the machine, take the ball out and go for the and take a sample of that particular granule and go to the QC lab to detect the LOD. How AI integration can be used in this way? How we can save the time of operator and save the time of company by using AI. In AI, what we do is there is continuous moisture analysis being done on using AI sensors of all the granules. Whenever once the required moisture level is attained during the fluidization of the granules, the system automatically stops the machine and at the required LOD. So this leads to the saving of a lot of time. In wet and dry granulation, the goal is to hit the precise endpoint, that is optimal granule size and moisture without over-processing. NIR sensors enable real-time AI-driven control. AI-driven endpoint detection, instead of stopping the machine, just like I said, on the fixed timer, like 10 minutes of drying, the AI continuously monitors the spectral signature of the granule bed. Once the target profile is matched, like the LOD, once the required LOD is achieved, the panel stops automatically, eliminating batch-to-batch variability and over-processing risk. AI in tablet compression. The modern tablet processes are data region environment. AI transforms this data into real-time quality control and predictive maintenance. Weight and hardness control. We have advanced machines in our pharmaceutical industry. Suppose we are compressing a tablet at 100 mg, right? If there is a disruption in the flow of the granules from the hopper, sometimes it happens is that tablet which is compressed is of 110 mg or it can be of 90 mg. How AI-driven controls can be used? AI continuously monitors the weight of each of the tablet being ejected from the discharge tube, and if AI detects there is out of limit tablet being processed by the machine, it automatically rejects the required tablet. So it leads to the prevention of market complaints. Weight and hardness control. AI adjusts the feeder speed and pre-compression force in real time. Punch monitoring. Due to overuse of punch, sometimes it happens is that there is a capping problem which occurs in the tablet. How AI can prevent this capping problem? Vibrations and acoustic sensors detect punch, tear or chipping before it causes weight variation or capping. Quality control. If AI detects uncontact uniformity, drift, the API isn't, that is the API isn't reached properly, it diverts those specific tablets to the rejection bin. Just a second. AI film coating. The coating uniformity and endpoint determination have traditionally relied on weight gain. Actually, what happens in coating is we continuously keep on coating a tablet until the required weight gain is achieved. The weight gain achievement is the endpoint of coating. But how we can use AI in endpoint detection in coating without even stopping the machine? Endpoint determination. Raman spectroscopy or NIR probes, they monitor coating thickness in real time, eliminating overcoating and reducing cycle time. Spray pattern analysis. AI monitors nozzle performance and atomization pressure to ensure uniform coating distribution across the tablet bed. Defect detection. The system in coating it is fed with a perfect picture of a tablet. Like the tablet is round, the tablet is having a bisecting line on one side and plane on other side. The system is fed with this type of gold standard to it. And whenever the AI detects that during coating, this type of tablet is not being matched with the gold standard, the AI rejects all those coming tablets. Defect detection. Computer vision inspects tablets in real time for picking, sticking or orange peel effect, triggering immediate parameter adjustments. And end-to-end AI coverage across unit operations. AI integration spans the full dosage manufacturing chain from granulation endpoint detection and through to the final packing, creating a unified data-driven quality layer across every critical processing step. However, there are bottlenecks to use of AI because on 2nd of April of this month, there was a FDA 483 observation being given to one of the companies in Michigan. What they were doing was they were just writing a prompt to ChatGPT or whatever for software they must be having and creating protocols and DMRs. So there was a 483 observation being given to them because AI cannot, yes, AI can help us in creating the protocols, but human intervention is needed. The key adoption barriers are validation complexity. The FDA 483 observation frequently cite insufficient AI model validation documentation and unclear change control procedures for redundant algorithms. The data integrity. The legacy equipments lack the sensor infrastructure and data pipelines required to feed reliable training data into the AI models. And the regulatory ambiguity. Evolving FDA guidelines on AI and machine learning in manufacturing creates uncertainty around what constitutes as acceptable control strategy. AI and the IPR strategy. Innovation in 2026 is no longer about finding a needle in a haystack. It's about using AI to build a magnet that brings the needle to us, while our IPR strategies ensures we are the only one holding the magnet. AI and the way forward. AI doesn't replace the quality manager. Even yesterday we were talking about that AI will lead to a loss of so many jobs in the pharmaceutical industry or even AI. Yes, there are some cases that AI has, you can say particularly in the IT sector, AI has taken up some of the jobs, but in pharma there is no such case because AI doesn't replace the quality manager. It gives them a superpower because it turns 10 years of dusty paper archives into live breathing advisors that prevents the mistake before it happens and increases the productivity. It is a garden of patient safety, it is an architect of resilience, and it is a catalyst for innovation, just like Dr. Neils was saying just before that. So, with that I close my words and the questions please from the audience.
So, any more question from the participant? I have a question, sir. How AI can be used for smart packaging material for enhanced productive life? Can be used for packaging material as well?
Sir, yes, we are using, right now we are even using AI in packaging. Suppose you have a blister and there is a printed aluminum foil side and the leading side. The printed side, it contains a pharma code. Pharma code is a unique combination of plus and minus and they give a particular value to you, right? And the count of that pharma code is suppose 23. There is a particular count of that pharma code. AI sensors are installed on the machines and it continuously see for that particular pharmacode on that machine and assures that only that particular printed aluminum foil is being packed for that particular blister.
So it was a pleasure to listen on AI for enhanced productivity. In the era of Industry 4.0, AI is playing a transformative role by involving more faster, smarter, and efficient production system, engaging quality as well. Dr. Sharma has shared the significance of real-time data and waste minimization as well. So today we have an excellent application of AI for product and multiple applications as well in pharma industry. So I congratulate and thank you. Thank you, sir.
Thank you, sir, for sharing your words of wisdom. And thank you to Dr. Chandan Mohan for concluding the session. Now I request Dr. Chandan Mohan to felicitate our speaker, Dr. Sandeep Sharma. Give a round of applause. Thank you, sir. Exploration ignites excellence and collaboration creates innovation. With this, let us continue our journey of knowledge and scientific exploration. We are honored to have this session chaired by Dr. Chandra Mohan again, and we want him to introduce our next plenary speaker, Dr. Krishna Kumar Chaudhary, Department of Botany, Banaras Hindu University, Varanasi. So please grace the dais.
He is currently working as assistant professor in Department of Botany, Banaras Hindu University, Varanasi. He is also served as an assistant professor in Department of Botany, School of Basic and Applied Sciences, Central University, Punjab, for more than 7 years. After completing his BSc, MSc, and PhD in Botany from BHU, Varanasi, he has worked as a visiting scientist in Institute of Soil, Water and Environmental Sciences, Agriculture Research Organization, Ministry of Agriculture and Rural Development, Israel. He has also worked on in some visiting scientist, NIPGR, New Delhi. Dr. Chaudhary has also delivered several talks at various international and national conferences and seminars, and also won some awards on national and international level. He is a life member of some prestigious Indian scientific societies. Dr. Kumar will speak on evaluating salinity stress modulated capsaicin for therapeutic applications against prostate adenocarcinoma, ADMET network, pharmacology, and molecular dynamics insights. Over to you, sir.
Thank you, sir, for the nice introduction. Good afternoon to all, and thanks to all the organizers for giving me this opportunity to be the part of this conference. Here, the main motto is to just show that basically the salinity stress throughout the globe is causing severe problem in productivity of the crops as well as severe soil complications. So basically the world population is expected to by the 9.7 billion that is used by 2050, and we need to basically focus how to feed this huge population. But on the other hand, due to the global climate changing scenarios, we are losing toward the productivity loss. And the World Soil Bank is also indicating that the salinity level is increasing by 10% each year, and it is severe condition and an alarming rate for us. and as per the economic loss, we can say that it is around 31 million US dollar per year. So after that, when we see about the main focus here, what is the purpose of the study? So basically, FAO Food and Agricultural Organization has given some 4 major steps how to overcome these saline activities. So here, the first step is the cultivation of salt-tolerant varieties of vegetables and crops, including the medicinal plants also, and then adaptive farming strategies adapted to salt-affected areas, and the sustainable utilization of saline soils and water resources, along with the restoration of health of saline soil and water through nature-based solutions. Here, my present work, which I will show here, that is mainly compiling about the 3 objectives of the FAO, and the 4th objective we are working on it, but not presented in this work. First of all, you all are aware with the capsicum, the chili, and it has several medicinal properties, for example, like anti-cancerous, anti-obesity, anti-inflammatory, and several other perspectives. So, these, because we see the Indian scenario. In India, basically India is the highest consumption as well as the producer as well as the exporter also of the chili plants. So basically in Indian kitchens, we used to use these chilies every time, and this could be a possible medical purpose also, yeah, medicinal perspective fulfilling. When we see about the data of the chili, India is among all the countries, India is the major exporter, producer as well as the consumer. And further, the study focuses upon the impact of the salinity on the chili cultivars, along with to identify the sensitive as well as the resistant cultivar about the salinity stress. Further, we have also utilized the cumulative stress response index so that we can classify the varieties of the cultivars under it, and further HRMS study for the metabolomics as well as some KEGG expression analysis, docking and molecular dynamic simulation have been done. This is just the morphological features among the saline structures, and when we see the salinity level above 50 and 100 millimolar have severely affected the plant growth throughout, and after that when we see the root length as well as the shoot length under the salinity stress. Although 100 millimolar salinity stress is causing more severe damages to the plant as compared to the 50 millimolar salinity stress. So moderate salinity stress could be a possible tool. We can harvest maximum medicinal compounds from the medicinal plant, as well as here the capsaicin is one of the important medicinal compound we extract from the chili plant. And these are the harvest. After harvesting, we have seen the reduction in the yield also, and after that we have also done the plant various morphophysiological parameters like reduction in plant growth, shoot length, root length. Along with that, we have also assessed the photosynthetic capacity and PS2 of the plants, and all of them are severely damaged, and the reduction have been observed, tremendous reduction have been observed, and the reduction is significant. And after that, we have just seen the chlorophyll, carotenoid and other pigments. They are responsible for basic survival of the plants. For example, chlorophyll and carotenoid is mainly leading towards the photosynthetic capacity of the plants. And after that, we have also done total phenolics as well as the total flavonoid content, which is better as the antioxidant for survival of the plant in stressed conditions. Then we have also done the reactive oxygen assessment of the reactive oxygen species, mainly causing oxidative damage to the plant cells and membranes. And after that, we have also done the several antioxidative enzymes, how the antioxidative enzymes have been stimulated. Based on all these parameters assessed under the 50 millimolar as well as the 100 millimolar of salinity stress, we have done the cumulative stress response index, and we have taken 4 cultivars. They were developed basically from IIPR, Indian Institute of Vegetable Research, Varanasi, and we have just observed that Kasi Gaurav was most resistant as well as the Kasi Ratna was most sensitive cultivar among the chili cultivars. Part of this study has been submitted to plant biology journal. The ultrastructural responses have also been noted on the leaf structures of the sensitive cultivars, and we have observed several defects among the stomatal apertures, smaller sunken stomata, as well as the number of stomata have been reduced. And the impact was most high in 100 millimolar. We have done the HRMS study, and the peaks are clearly suggesting the shifting of metabolites under the 50 millimolar as well as the 100 millimolar. However, the valuable peaks were observed near under the 50 millimolar. And then the enrichment analyses have suggested that the sensitive cultivars among the 50 millimolar or higher, when we are talking about the salinity stress 50 millimolar and 100, under the 50 millimolar stress, the phenyl-propanide pathway is more upregulated, that is suggesting for the protection of the plants. However, under the 100 millimolar, the amino acid metabolism is more upregulated, suggesting that the plants are struggling for their survival. And the part of this have been published in the food chemistry. Now we are very much new in the bioinformatics analysis. We have taken some bioinformatics approach also with the capsaicin. The capsaicin biosynthesis have been observed 1.634 log to fold changes higher among the 50 millimolar, while it is about 0.33 only under the 100 millimolar. So this capsaicin, we have also performed the boiled egg illustration, and the capsaicin is well lying between the boiled egg model and also under the drug likeness criteria for representing the molecules. Further, we have done the target net as well as the Swiss target net predictions, and out of all these, we have found that 65 are the common, and both suggested that 23 are main target they are affecting, and they are responsible for like cytochrome P450 and other phosphatase activities. Then the PPI network clearly suggesting the identification of top 5 hub genes. Basically, these are 5 hub genes. They are mainly top 5. And further, we have done the KEC pathway enrichment analysis. They are also suggesting that the capsaicin is easily and strongly binding towards the molecules. Then we have done the KEC pathway network illustrated that potential role of hub gene is the development of progression of the prostate cancer, and the red colors are highlighting the pathways where target pathways where they are involved. And these are some of the 3D structures of the these 4 hub genes we have identified. They are playing major role in the adenocarcinoma cancer treatment. We have done several RNA expression analysis, which is also suggesting that these genes are clearly playing a stronger role in the treatment of the cancer. While when we see the docking results also for each and every capsaicin hub target proteins. And further, we have said upon the molecular docking analysis, we observed that the capsaicin is clearly binding affinity with 6.720, and it is the strongest binding affinity with PIK3CA. Then the molecular dynamic simulation also clearly suggesting the Eisen value lies between the, and they are following the Lipinski rule, and further for each like PRBB2 and then HSP0 AA1, similar the Eisen value for the mTOR and the PIK3CA. And these all the bioinformatics studies have been published in silico pharmacy. pharmacology. Now just concluding to this, basically when we utilize 50 and 100 millimolar of salinity stress, we have observed that we have harvested maximum capsaicin content under the 50 millimolar as compared to the 100 millimolar. And also when we have screened our cultivars, we have observed that the Kasi Ratna was highly sensitive and the Kasi Gaurav was the least sensitive under the salinity stress. And the USDLC HRMS study clearly states that the moderate salinity stress can be utilized as a potential tool to stimulate capsaicin biosynthesis in capsicum. And ADMET properties are also showing the favorable drug likeness, exhibited high gastrointestinal absorption, blood-brain barrier, permeability, and as well as the good solubility. So PPI analysis identified 5 half genes and then they are enriched in cancer-related pathways, and further 4 half genes they were directly involved in the prostate cancer pathway. And further in the expression analysis we have observed they confirm the clinical relevance of the oncogenic drivers in these. And the molecular dynamic simulation clearly shows the highest flexibility of HSP90AA1 for suggesting strong conformational responsiveness against the adenocarcinoma. And these are the work. Basically, I have shown the work here. Amalika Singh is working with me, the student involved in this capsaicin and the chili plant with salinity stress. And these are some of the funding agencies like ANR and the Institution of Eminence, BHU. And also, I would like to acknowledge BHU for Professional Development Fund and CDC for the analysis. Thank you.
Thank you, Dr. Krishna. Dr. Krishna has expertise on stress physiology, and he has spoken on network pharmacology, ADMED, and molecular dynamics. This area is of utmost importance in present scenario, especially in the area of pharma world. So, thankful Dr. Chaudhary for introducing such an important use of capsaicin in therapeutics, the active oxygen species role, and how they are significant for salinity stress modulation against prostate disease. So, thank you so much, sir. Thank you.
Thank you, sir, for sharing your idea of innovation and excellence. Thank you. Now I request Dr. Chandra Mohan to felicitate our speaker, Dr. Krishna Kumar Chaudhary, for his wonderful session. Thank you, sir. Next, I request Dean SMAs to felicitate Dr. Chandra Mohan for sessioning the chair. And please. Thank you, sir. Thank you, ma'am. Now I request all our dignitaries and guests to proceed for lunch. First, our guests and dignitaries will proceed. Students, please be seated. All students, please be seated. First, our guests will proceed.
आ जाओ जल्दी से इधर।